मजबूत कर देते हैं। जो लोग इंसानियत के लिए काम करते हैं, जो चीज़ भी इस कायनात में जितनी भी चीज़ें हैं, जो जो चीज़ें इंसानों को फ़ायदा पहुंचाती हैं चीज़ें, अल्लाह ताला उनको मजबूती अता करता है और उनको जमा देता है वहां पर कि कोई हिलाना चाहे तो हिला नहीं सकता उनको। ये तो इस आयत का निचोड़ और खुलासा है। अब आप ये देख लीजिए कि इस पूरी कायनात का जो असल है वो दूसरों को फायदा पहुंचाना है, दूसरों से हासिल करना नहीं है, दूसरों को देना है। इस पूरी कायनात का आप रिलीजन कह लीजिए अगर, कि अब पेड़ को ले लीजिए, पेड़ जमीन से क्या क्या चीजें लेता है, पानी लेता है, खाद लेता है जमीन से, और जमीन के अंदर जो खास चीजें होती हैं वो पेड़ लेता है। सूरज से गर्मी ले लेगा, हवा से नाइट्रोजन ले लेगा, फिर उसके बाद वो पेड़ अपने आप को तैयार करेगा। इतनी सब चीजों के लेने के बाद फिर पूरी जिंदगी वो देता ही रहता है लोगों को, कभी साये की शक्ल में, कभी फल की शक्ल में, कभी फूल की शक्ल में। फिर बेचारा जब वो खत्म हो जाता है तो आपके और हमारे जलाने के काम में आ जाता है, यानी उसकी पूरी जिंदगी दूसरों को फायदा पहुंचाने के लिए। ये पहाड़ है, ये इंसानों को फायदा पहुंचाने के लिए, ये दरिया बह रहे, ये दरिया बह रहे हैं, ये नदियां हैं, ये पानी के जितने भी आबशार हैं, ये सब इंसानों को फायदा पहुंचाने के लिए। सिर्फ एक इंसान ही बचा है जो दूसरों से फायदा हासिल करने की कोशिश में लगा रहता है, देने के लिए उसका जहन जल्दी तैयार नहीं होता, बस वो सिर्फ हासिल कर ले किसी न किसी तरह के, कुछ ले ले किसी से। ये कायनात का इस नेचर का जो उसूल है, ये उसके खिलाफ है। दीजिए, जो कुछ भी आपको दिया है उसने, आप दूसरों के लिए फायदा पहुंचाने की कोशिश कीजिए। अब इसकी दो मिसालें, दो नाम आप ले सकते हैं। एक हमारे हकीम साहब, पूरी जिंदगी जिन्होंने सिर्फ इंसानों को देने में गुजारी, जो कुछ भी वो सोचते थे और उसको वो दूसरों को देने के लिए सोचा करते थे। और दूसरा नाम आप सर सैयद साहब का ले सकते हैं, जिन्होंने इतना बड़ा इदारा अलीगढ़ मुस्लिम यूनिवर्सिटी कायम किया। तो कहने का मतलब ये है कि जो लोग इस उसूल को समझ लेते हैं कि हमें देना है, तो अल्लाह उनके पैरों को मजबूत कर देता है। हकीम साहब आज दुनिया में मौजूद नहीं है, सर सैयद भी आज दुनिया में मौजूद नहीं है, या इन जैसे और लोग जो हैं वो हमारी नजरों के सामने नहीं है, लेकिन आज भी जब उनका नाम आता है तो हम सब का सर झुक जाता है, मोहब्बत के साथ और अकीदत के साथ, कि उन्होंने खुदा के उस उसूल को आगे बढ़ाया कि देने वाले बनो तो तुम्हारे पैरों को मजबूत कर दिया जाएगा। और आखिरी बात कह के मैं अपनी बात खत्म करूंगा। मैं मस्जिद में इमाम हूँ, दुनिया के तरह के लोग आते हैं और हर मिजाज के लोगों से वास्ता पड़ता है। बहुत सालों पहले की बात है कि मस्जिद में रोजा खोलने के वक्त एक आदमी आता था और बहुत से आदमी, सैकड़ों आदमी लाइन से बैठ के रोजा खोलते थे रमजान के महीने में। एक आदमी आया करता था, बूढ़ा बिल्कुल, और वो एक थैली में हर रोज इफ्तार के वक्त कुछ लेके आता। और वो जानते हैं आप उसकी थैली में क्या होता था? मुरमुरे। मुरमुरे मुरमुरे समझते हैं आप सब लोग? मुरमुरे लाता था वो, और एक एक मुट्ठी मुरमुरा वो सब रोजेदारों के सामने रख देता था, और पूरे रमजान कई साल तक वो लोगों को मुरमुरे खिलाता रहा। मैंने उससे एक दिन पूछा, भाई ये आप ये काम क्यों करते हैं? कहने लगे कि मैं सड़क के ऊपर केला बेचता हूँ और हर रोज उसमें से कुछ पैसे निकाल कर मैं रोजेदारों के लिए रख देता हूँ, और रोजेदारों के लिए इन पैसों से मुरमुरे खरीदता हूँ, फिर इनसे लोगों को रोजा खुलवाता हूँ। मैं रोजा इफ्तार करते हैं लोग मेरे इन चीजों से। कुछ दिन के बाद वो बूढ़ा खत्म हो गया, लेकिन आप जानते हैं कि उसके खत्म होने के बाद बरसों लोग उसके लिए इफ्तार के वक्त दुआ करते थे, बरसों। क्यों? वो इंसानों को फायदा पहुंचाने के लिए, उनको रोजा खुलवाने के लिए अपनी पाक और हलाल कमाई उनके ऊपर खर्च करता था। फिर लोग उस बूढ़े को याद करते थे। जमाना गुजर गया, आज भी उस जमाने के जब लोग इफ्तार करते हैं तो कभी कभी कहते हैं कि वो मुरमुरे वाला बूढ़ा कहाँ गया। तो मैं आप सबसे गुजारिश करूंगा, आप सब यहाँ से भी कुछ ले रहे हैं, जामिया हमदा से भी, और स्कूलों और कॉलेजों से भी आगे आइंदा आप कुछ लेंगे। इन सब के बाद आपका सिर्फ एक मकसद होना चाहिए कि इंसानों को फायदा पहुंचाएं, जैसे कि इस कॉन्फ्रेंस का मकसद है।
Thank you, Imam Sahab, for such enlightening words. May Allah guide and protect us and reward you abundantly. The Department of Biotechnology organizes such conferences to enlighten the young minds and promote the participation of students at all levels. But because of the pandemic and the critical situations we all faced in recent times, the department is organizing this event after 3 years. So I take privilege to bid a very warm welcome to all the renowned speakers and delegates who took out their valuable time and joined us today to be part of this conference. We are honored to have you all with us. This year, the theme is based upon the theme of sustainable development, which is the pathway to the future we want for all. This conference will help the students to understand how biotechnology can be applied to address the global challenges we are facing. This conference will also allow us to meet some of the brilliant minds of the country, so I guarantee that the conference will be productive and worth your precious time. Now, moving ahead towards the program, I am profusely elated to take an opportunity to welcome our Honorable Chief Guest, Professor Asim Ali Khan, Sir, Director General of Central Council of Research in Unani Medicine, Ministry of Ayush, Government of India. Thank you, sir, for accepting our invitation and taking out some time for us from your busy schedule. Sir hardly need any introduction. He has made all of us proud of his distinguished work in numerous capacities. He is one of the most celebrated dignitaries, the man of distinct vision and a fountainhead of illuminating ideas, an idol of knowledge and experience, and he is an inspiration to all of us. Now, I would like to request our Honorable Vice Chancellor, Sir, to please welcome our respected Chief Guest, Professor Asim Ali Khan, Sir, with a planter. Now, thank you so much, sir. Now, I would request respected Dean, Professor S. Raisuddin, sir, to please present the planters to Honorable Vice Chancellor, sir, Professor M. Afshar Alam, who has been working immensely in achieving the mission and core goal of this great institution. With the leaders like our Vice Chancellor, sir, the proverb like action speaks louder than words seems to do justice. Now, I request Professor M.Z. Abdin, sir, Head Department of Biotechnology, the organizing secretary of this event, to please offer the planters to Professor Raisuddin, sir, Dean SELS, who is constantly working towards connecting the young minds of the school to strive towards success and achievement. Now, I request Professor Raisuddin, sir, to please present the planters to Professor M.Z. Abdin, sir, Head Department of Biotechnology, who is known for his wisdom and knowledge, the man who is tirelessly working in taking the department towards the new heights. Thank you, sir. Now, moving ahead, I would like to invite all the members of Tarana team to please be on stage for the University Tarana.
ये दर्स गहे हम दर्द है 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 हम इसके चांद सितारे हैं ये दर्स गहे हम दर्द है ये दर्स गहे हम दर्द है ये दर्स गहे हम दर्द है ये ताबीर है ये उस ख्वाब की जिस ख्वाब की मंजिल हिकमत है जिस ख्वाब की मंजिल हिकमत है ये शम्मा ऐसी शम्मा है जिसकी के जहां में शोहरत है जिसकी के जहां में शोहरत है इस वक्त गुजर रहे हैं हम सब पर तारे बनकर चमकेंगे मानिंद गुलों के महकेंगे महताब की सूरत चमकेंगे। ये दर्स गहे हम दर्द है ये दर्स गहे हम दर्द है ये दर्स गहे हम दर्द है ये। नुसरत व ज़फ़र का परचम हम इस आलम पर लहराएंगे। इस आलम पर लहराएंगे। इस रोशन शम्मा की खातिर हर तूफान से टकराएंगे। हर तूफान से टकराएंगे। सरसाज रहे शादाब रहे अल्लाह दुआ ये मेरी है। इस गुलशन पर नावा जाए अल्लाह दुआ ये मेरी है। ये दर्स गहे हम दर्द है ये दर्स गहे हम दर्द है ये दर्स गहे हम दर्द है ये।
Thank you, Tarana team, for the heartfelt University Tarana. Listening to it makes us proud every time to be part of this cultural milestone called Jamia Hamdard. Now, without any further ado, I would like to begin with the scheduled event of the day by inviting Professor Raisuddin Sir, Dean School of Chemical and Life Sciences, for the welcome address. We need not to say much about Sir, as the whole campus is already vibrating and echoing his prominence. Sir has proved his excellency as a Director of Research and Development Cell, Jamia Hamdard, and now relentlessly working as a Dean of School and Chemical and Life Sciences. Welcome, you, sir.
Thank you. Assalamu alaikum and a very good morning. Honorable Vice Chancellor, Professor Akhtar Alam Sahab, the Chief Guest of the program, Professor Asim Ali Khan, Director General, Central Council for Research in Iranian Medicine, Ministry of Ayush, Professor M.J. Dati, Head of the Department of Biotechnology, and all the invited speakers, delegates, students, and research scholars. It is a very proud moment for our school that the Department of Biotechnology is organizing a very relevant and pertinent conference on the role of biotechnology in sustainable development and human development. And you know that UN has declared 17 SDGs, they are called Sustainable Development Growth. And when you see all these goals, you will find that biotechnology has some role to play in many, for example, health, agriculture, environment, and education. The 4-5 goals are there where biotechnology has some role. For example, sustaining humans for their medicine, for their food, and agricultural things. So, biotechnology has quite relevance in the many sustainable development goals which are many countries are following. So all the talks in scientific community, politics, as well as in other sectors is surrounding around the SDGs. And this even recently COP27 was adopted there also, that is aimed at sustainable development. And a country like us, where many indices of human growth and development are not so good, recently hunger index or happiness index, many things India has not fared very well. So because of if we apply new technologies, including biotechnology, artificial intelligence and machine learning, things like that, many countries which are adopting such technologies are very far ahead in terms of those indices which recently has been released. So I hope that this 2-day event on today and tomorrow will be very much discussing on these issues of the sustainable growth, which where biotechnology has to play some role. Department of Biotechnology of School of Chemical and Life Sciences is the flagship department. The courses at undergraduate level, postgraduate level, and doctoral level are very sought after. The maximum number of applications which are received for any course in this our school are for the biotechnology. Biotechnology is the department whose courses are seats are filled very early, and many people seek. And the first priority, though I'm a toxicology head and also the department of toxicology, I envy that most of the students give first preference as their course choice to the biotechnology courses, UG or PG. And the seats which are there in biotechnology are also all the teachers where have some projects either sponsored by the government agencies, SERB, DBT, CSIR, ICMR, all major funding agencies, including this is also the department which is supported by UGC, in Fish Department, DST, UGC, the SAP, too. So all the major distinguished teachers which a department can achieve has been achieved. And you will be surprised to know this is the newest department of our school. In the beginning, in 90s, when the school was established as the Faculty of Science, that time it was part of botany. So it bifurcated sometime around, I think, 1999, if I'm correct. Then, so the journey of this department is very quick and fast. The growth attained by this department is very spectacular. So I congratulate the organizing secretary, Professor M. Dept, for conceiving this program for 2 days, and I wish that you all visit the department. Department has a special center for transgenic plant development and focused on transgenic development. Department has many patents also, which have been commercialized also, especially focusing on artemisia. And recently, animal biotechnology has also taken good lead in this department. Many new faculties who have joined, they are from the animal biotechnology field. So I hope that you will find this conference very useful. I welcome all the delegates from this Delhi or outside of Delhi, very premier and reputed institutions, including from Pune University, our guest is there. So I hope you will listen from them through the oral presentation and poster presentations by the young scientists, and it will be a good opportunity for you to know how biotechnology is progressing. So I don't need to talk about much importance of biotechnology. Biotechnology is there. It is a relevant subject for all human activities: health, medicine, agriculture, environmental biotechnology, medical biotechnology, and agriculture biotechnology. And all the time, this subject is also new in news also. Currently, GM crop and artificial meat has been synthesized. So, a lot of debate, moral debate, is also on the biotechnology. So, I hope that these 2 days event will be very much useful to you. You also visit the department if you have time, and the department has also opportunity to take you there. See the facilities. The all major facilities are there, including the confocal microscopy there, right in the department. Also, we have transmission electron microscope. Only 5 or 6 institutions in Delhi are functional transmission. It is there in pharmacy faculty, but all research scholars can use it by paying very nominal amount, and you can also see the facilities, all major research facilities. And therefore, our department, our university is ranked #1 in pharmacy, and biotechnology is also contributing to that ranking. So I again thank the Honorable Vice Chancellor and the Chief Guest for their time which they have spared for this program, and congratulate Professor M. Dept and his very vibrant, young and enthusiastic team for making this. And I know that this program was organized within 1.5 months only. So with this very short time with them, they have made a very good preparation for you. And please be part of this conference very vividly and enjoy your presentation at this very beautiful campus that is Jamia. Thank you very much.
Thank you so much, sir, for your kind words. Now I would request Professor M.Z. Abdi, Head Department of Biotechnology, the man behind organizing this event, who, without minding his health, shows a keen interest, for the introductory remarks.
Bismillahirrahmanirrahim. Assalamu alaikum. Good morning to all of you. Let me thank Professor Afshar Alam Sahab, our Honorable Vice Chancellor, and Professor Asim Ali Khan Sahab, the chief guest of today's event, for coming so early on our invitation to grace this occasion. I also thankful. I am thankful to our team for coming to this event. Actually, all the delegates, deans, head of the departments, and dear students. Today we are going to have 2 days seminar, which is beginning from today, and it will culminate tomorrow. This is on role of biotechnology in sustainable development and human welfare. So, as we already aware that United Nations has in 2015 has set 17 SDGs for the world to follow, so that we can wipe out hunger, poverty, malnourishment, and alleviate suffering of human beings from health and diseases. In addition to that, actually, the goal which is set forth is to provide clean air, clean water, and also we have to purify our earth, which is also quite highly polluted, and that ultimately culminate into providing food that is also contaminated. So actually, with these aims in mind, we have organized these 2 days seminar, national conference, and actually our delegates are from different parts of the country. We have invited eminent scientists, and hope that with these 2 days events, we will be able to come out some resolutions and solutions to achieve the goals set forth by United Nations and ratified by our country and the global leaders. So we will work for serving not only the people from our own country, but around the globe and the planet Earth also. Okay. So with these words, I am handing over podium to Monica. Thank you very much.
Thank you so much, sir. Thank you for introducing us to the theme of this conference. Now, I am profoundly delighted to have this opportunity to introduce the Honorable Chief Guest for the day, Professor Asim Ali Khan, sir, the Director General at Central Council for Research in Unani Medicine. He is a renowned scientist of Unani medicine and has a vast experience in clinical medicine research and teaching. He and his team are actively engaged in a standardization program and developed pharmacopoeial standards for 298 single drugs and 100 compound formulations which have been published in Unani Pharmacopoeia of India. He is responsible for the development of infrastructure, new campuses of regional and national research institutes of Unani medicine and medical centers. Recently, he and his team played a key role in the development of first regional research institute of Unani medicine in Silchar. As a member of National Commission for Indian System of Medicine, he is involved and responsible for policy making, planning, implementation, and governance of quality education, training and research in Ayurveda, Unani, and Siddha sciences. He has played. Just one line, sir.
Yes, please.
I am feeling honored to mention that he also served as a professor in Jamia Hamdard and actively participated in many academic and administration functions for about 30 years. We are honored to have you here, sir.
Assalamu alaikum and a very good morning to all of you. Of course, indeed, I am from Jamia Hamdard. I've been working here in almost all the capacities, other than even as a professor. And I feel that all the students, when I'm seeing more of the students are there, other than my colleagues, you are very lucky to be part of this organization, because I know this organization in and out. We together, the colleagues who are on the stage and the colleagues who are down, sitting there, we really have worked hard together to make this institution what you are seeing it today. I feel that, I can really feel that you all are so lucky to be part of this family, extended family, and I wish every best to all of you for the institution. Very Honorable Vice Chancellor of Jamia Hamdard University, Professor Abshar Alam Sahib, the Dean of the school, Professor Veis Sahib, and the Head Department, Professor Abdeen Sahib, my colleagues from Jamia Hamdard, distinguished delegates and dear students. It is indeed a privilege for me to be here today and address this August gathering on the inauguration of this two-day national conference on biotechnology for sustainable development and human welfare, organized by the Department of Biotechnology, School of Chemical and Life Sciences, Jamia Hamdard. You all know that this school is well known for, and all over India, for its state-of-the-art infrastructure and for cutting-edge research in the area of biochemical and life sciences. I know many students, when we go to states or some other parts of the world to travel, and we find the most of the people which we find outside the country in the best organizations across the globe, they are from the Department of School of Sciences, here, Life Sciences, or Pharmacy. And many of them are in the wonderful positions outside the country. I am very happy to know that the two-day conference will be focusing on cutting-edge technologies and ongoing research in the field of biotechnology to achieve the United Nations Sustainable Development Goals. This Department of Biotechnology, the School of Chemical Life Sciences, has an international repute, as I told, with a strong interdisciplinary research and teaching base in plant and animal biotechnology, with active collaboration of industries and healthcare institutions. It is certainly well positioned to make a significant contribution in achieving the Sustainable Development Goals that embody ecological, social, and economic requirements. As we all know, that the biotechnology is the life science which generally deals with the study of living organisms. It is an application of biology and other different techniques to change or modify products for specific human use. Biotechnology plays an immense and important role in human welfare and has revolutionized the mankind since its existence. It contributes much towards the human welfare and their health needs. In the field of medicine, if I talk in particular, biotechnology is being widely used in the development of several innovative techniques for diagnosing, treating, and preventing diseases. It helps in providing effective treatment and prevention measures for different diseases through its intervention of novel drugs and recombinant vaccines. With the help of modern technologies and biotechnologies, many diagnostic tools have been devised to detect the diseases in a quick and accurate manner, and that credit goes to our colleagues, the biotechnologists. The important role played by the traditional systems of medicine in meeting the global healthcare needs has been universally acknowledged now, and after the COVID, it has been more so, it has happened that way. Today, we are exposed to a range of communicable diseases, lifestyle disorders, nutritional issues, and medical care problems. The incidence and prevalence of diseases, complications, and the impact on the quality of life of patients and family members may be minimized to a significant extent if quality assured healthcare services, including traditional medicine services, are accessible to everyone. Our country is privileged being home to time-tested indigenous medical systems, that is the Ayush systems of medicine, which are being practiced here for centuries. The Unani system of medicine, which is an integral part of Ayush and the Government of India's public health initiatives, is a comprehensive medical system that provides preventive, curative, and rehabilitative healthcare. The Government of India has been providing increasing patronage and support and funds for its multifaceted development of Unani medicine, while it is also providing to other systems of medicine, including allopathy. And as a result, the Unani system of medicine today, not only in India, is the world leader in terms of the Unani system of medicine. We are the world leaders as India. Unani medicine offers treatment for disease related to all the systems and organs of the human body. The treatment for chronic ailments, skin disorders, disease of liver, musculoskeletal and reproductive systems, and immunological as well as lifestyle disorders have been found to be highly effective and acceptable. The Central Council for Research in Unani Medicine is the apex organization for research and development in Unani medicine. Over the years, which is more than 4 decades, the CCRUM has emerged as the leading government organization for scientific research in Unani medicine. The areas of research include clinical research, including safety and efficacy studies of Unani drugs, validation of the fundamentals and research therapies and regimental therapies, drug standardization and quality control, survey and cultivation of medicinal plants, and literary research. The Council is conducting researches on fundamentals of Unani medicine with the objectives of providing scientific basis to the fundamentals of Unani medicine and validating the theories of fundamental research in the light of genetic, biochemical, physiological, and pathological parameters, and thereby correlating and trying to correlate the theories of fundamental research with modern biology and biotechnology. 20 such projects on fundamental research have so far been completed by the Council's one of the national institutions that is located in Hyderabad. 6 studies on genetics and pharmacogenomics on theories of humors with special reference to diabetes mellitus and essential hypertension, as well as hepatitis and vitiligo, as well as cancer in healthy volunteers and patients are in the progress. And we are also in the process of developing a dedicated center for the fundamental research on biotechnological lines in one of the institutions in Hyderabad that is already in the process. The Drug Standardization Research Program of CCRUM has developed pharmacopoeia standards for single and compound formulations for incorporation in the Unani Pharmacopoeia of India. Besides standard operating procedures for method of preparation and pharmacopoeia standards, they are also being developed. And under this program, the CCRUM has developed the pharmacopoeia standards of more than 300 single drugs and around 200 compound formulations. Besides, there's a lot of new infrastructure and new buildings with the patronage of our leadership in the Ministry of Ayush, with the Minister of Ayush, Honorable Minister of Ayush, both ministers, and the Secretary as well as Special Secretary, under the able leadership of our Honorable Prime Minister, Shri Narendra Modi Sahib. We are working good way. A lot of support is coming to us from the government. Recently, we have added some very wonderful infrastructure. We have upgraded one of the central institution in Hyderabad as a national institution. One wonderful state-of-the-art facility has been, newly built facility has come in Mumbai, where we have shift inaugurated by Honorable Minister in July, in the month of July. We have been doing wonderful work, and day before yesterday, I was in Silchar, Assam, on 20th, just 2 days before. Is first of its kind and a very state-of-the-art building in Assam we have developed, and day before yesterday it was inaugurated by the Honorable Minister. It is almost 4 acres of land with a cost of almost 50 crore rupees that has been dedicated to the country, and more so on that particular region. We have issued one Center of Excellence grant to our center in Kashmir, and I am also very happy and pleased to again recall this. The Jamia Hamdard, we have been able to provide from the Ministry of Ayush as Center of Excellence in medicine and regimental therapy. Almost 3-4 months to come, and Jamia Hamdard is one of the Center of Excellence of Ministry of Ayush. I must mention here that the Jamia Hamdard and CCRUM have a long history of collaboration. There has been a regular exchange of experts and expertise between the two institutions. A lot of graduates and postgraduate scholars from Jamia Hamdard are already employed in CCRUM. An MOU has also been signed with Jamia Hamdard and CCRUM to work together in the areas of research and development of Unani medicine. The safety monitoring of herbal medicines, we all know that is an important concern nowadays. The assurance of the safety and efficacy of herbal drug requires monitoring of the quality of the product from collection through processing to the finished packages, product in the independent and dependent upon the standardization of these herbal drugs. Adverse events reported to the regulatory authorities in relation to the use of herbal products are often attributable to poor quality of source material and manufacturing and processing factors, among others. The herbal raw material is prone to a lot of variation due to several factors, and the important ones being the identity of the plants. Even when correctly authenticated, it is important to realize that different batches of the same herbal ingredients may differ in quality due to a number of factors such as inter or intra-species variations, time of harvesting, environmental factors, storage conditions, and processing treatments. Therefore, quality control and standardization of herbal medicines involve several steps like source and quality of raw materials, good agricultural practices, and good manufacturing practices. The sustainable supply of high-quality herbs is among one of the challenges of the health sector, and clearly there is an urgent need of alternative and complementary methods for uniform production of herbal medicine. In this context, the tools and techniques of biotechnology, like in vitro plant, cell, tissue, and organ culture, offer solutions in terms of mass propagation of plants. Biotechnology does have a high potential, not only to deal with the shortage of quality raw material in the traditional medicine sector, but also to deal with the new emerging diseases of complex nature. In addition, biotechnology should be considered as one tool in a larger portfolio of technological options to be applied where it is needed and when it offers the best available options for solving specific problems related to traditional systems of medicine. I'm happy to mention here the successful collaboration between Jamia Hamdard and CCRUM in the recent past, where with the Department of Biotechnology specifically, we have many collaborative researches going on in Jamia Hamdard with pharmacy, with Unani medicine, with science faculty, School of Sciences. One of the Department of Biotechnology, Professor Hamzad Abdeen Sahib, who is the head and professor of biotechnology these days, he is also the head of Center for Transgenic Plant Development, Department of Biotechnology. And this department, under his leadership and as a principal investigator, they have played an important role. And with the Jamia Hamdard Department of Biotechnology, we developed scar markers and a kit for the authentication of single Unani drugs. Many such studies are already going on in Jamia Hamdard. And the objective of these studies were to authenticate the species of plants through scar markers. And to give examples, they were like adulterant of Aftimun Vilayati, as well as Tochmikasus. And we have obtained patents on these, which have been worked by Dr. Abedin and his team in Jamia Hamdard. We have received patents on these scar markers and kit for the authentication of plants. So congratulations, Professor Abedin. Thank you so much for that. For the maximum benefits of the biotechnological applications in the Ayush sector, a meaningful dialogue among experts from traditional medicine and biotechnology sector is happening, and which is much needed. There is a lot of possibility and scope for collaboration between the Department of Biotechnology, Jamia Hamdard, and CCRUM, as well as other departments. As I told you, that it is pharmacy, it is science, it is some other departments which have come forward and they are doing good collaboration research, including Unani medicine. Professor Saeed and many other our colleagues now from pharmacy, I think 6 or 7 professors are there who are already working in collaboration with CCRUM. Before concluding, I would like to say a word of encouragement for my young students and researchers who are present here today. And this conference is a perfect opportunity for you. As I told you, you are in a wonderful place. Jamia Hamdard is a wonderful platform. I know this is a wonderful family. It has the foundation, as just now Farooqi Sahab was saying, it is a foundation and blessings of Hakim Abdul Hamid Sahab, Marhum Mahfoor, and they have really put a lot of sweat in this development of this. A lot of other people in Jamia Hamdard, or the leaders, they are working very hard, they have worked. And now I know Professor Afshar Sahib very well. He's doing wonderfully well. And he's a person under whose leadership he has a great skills and great learning from this organization, from outside. And I think this is, Jamia Hamdard is one of the fortunate organizations that we have Afshar Sahib in the form of the Vice Chancellor. And I hope that all of you are going to benefit with his leadership. Not taking much time, I once again congratulate the Department of Faculty in school and the Department of Biotechnology and colleagues, and also congratulate the students of this university, particularly from the science, that you are a part of this organization. Wish you every best, and wish every best for this wonderful conference. Thank you very much, and thank you so much for inviting me. Thank you very much.
Thank you, sir. Thank you so much for your words of wisdom. It will be a great pleasure for us to listen to you again in our future events. Now, I consider it a great honor to welcome our Honorable Vice Chancellor, sir, for his presidential remarks. As we all know, that sir is working immensely in achieving the mission and core goals of this great institute. Let me just mention that sir has organized various summits. I welcome you, sir.
Sorry, because I'm little bit hurry because we have some urgent meeting with UGC. Professor Asim Ali Khan, sir, the Director General of CCREM, Government of India, and today's chief guest of this function. Professor S. Raisuddin Saab, the Dean School of Chemical Science and Life Sciences. Professor N.J. Abdin Saab, the Head of the Department of Biotechnology, and other deans, colleagues, senior professors, and dear students. A very good morning to all, and I'm very sorry that we have preponed the function 1 hour before. So, the Department of Biotechnology was established in 1997 with a vision to be recognized as a department of international repute with strong interdisciplinary research and teaching based in plant and animal biotechnology with the active collaboration with industry and healthcare institutions. The department offers undergraduate, postgraduate courses and PhD with the excellent and updated curriculum as a blend of biological science and applied research. The teaching faculty in the department is very dynamic with the research experience in the lab of international repute, both in India and abroad. And their research is well recognized internationally. The students of biotechnology have a good rate of success in NIT, CSIR, UGC, DBT, JRF, ICMR, JRF, even the research associate for other funding agencies. They are also selected in reputed institute for PhD besides placement abroad. The department is a recipient of UGC SAP DST FIS program. In addition to that, the teachers are having their individual project grant from the various funding agencies such as DST SERB, ICMR, IU, CSIR, UGC, DBT, Welcome Trust, India Alliance, and also the consultancy project from the industry, which Professor Asim has already mentioned about the department relationship with the IU's ministry. The Center of Transgenic Plant Development as a unit of the Department of Biotechnology was established in 2004 with the research grant from CSIR, Department of Agriculture, Cooperation, Farmer Welfare. They are also coordinating with Agriculture Ministry, and they are getting a lot of industry grant from them. With the mandate of focused research in the development of varieties of crops, the medicinal plant with improved yield and quality of produce, as well as the development of DNA-based fingerprint of authentication of traditional medicines. Various labs in the center have a state-of-art facility to train PhD and postdoctoral students who carry out research in various disciplines in plant and microbiological biotechnology. Jamia Hamdard as a set of higher education learning recognition from across India and abroad. The ranking of Jamia Hamdard speaks itself. It stands 1 over the year in pharmacy or our management department, has also come into the top 100 level. The university remained in top 50 institutions in the country over many years, and in the Time World ranking, the Jamia Hamdard stands 601 to 800 bracket, with par for many central and state universities. In the Asian World Ranking, Jamia Hamdard stands 401 to 450 ranking. The World Southern Asian ranking is 110. So this is the great achievement considering this university as young one. And because of your research, this will be happen. With more innovative ideas, I do hope that Jamia Hamdard can live for top 25 institutions in the country in a couple of years and reach top institution, top 10 in the country over a year. in order to achieve the peace and prosperity for the people and the planet and the more sustainable future for all. In 2015, the UN General Assembly created a blueprint of 17 goals and 169 targets, which are collectively known as UN Sustainable Goal, that is SDG. And today's topic is biotechnology for sustainable development and human value. Is very important to that all 17 goals. Across the global challenge we face, including those related to poverty, climate, environmental degradation, prosperity, health, these goals drive the world of the UN agencies as well as NGOs and companies across the world. The researchers are now turning to biotechnology as a solution to achieve this goal. The Department of Biotechnology School of Chemical and Life Sciences is organizing this two-day National Conference of Biotechnology for Sustainable Development and Human Welfare. The recent advance in biotechnology help us to prepare for and meet the society's most pressing challenges. The biotechnology provide a breakthrough products and technologies to combat debilitating and rare disease, stop the outbreak of infectious diseases, reduce our environmental footprint, relieve poverty, feed the hungry, use less and cleaner energy, provide clean drinking water, protect biological diversity on land in our ocean, and we have safer, cleaner, and more efficient industrial manufacturing processes. This being a biotechnologist, we have the opportunity to encourage more innovation in biotech sector to achieve the UN Sustainable Goal. In this conference, the great visionary and thought leaders from different corners of the nation, which will well known of their thought of process to significant research work, are participating. The name few is Professor Deepak Pental, the man behind the genetically modified mustard, which is India's first transgenic food crop to be generated. Approval is going to enlighten us with the keynote address on the past and future of agriculture. There will be discussion on different aspects of biotechnology like sustainable agriculture, environment, as well as molecular and nanomedicine, infectious disease, and cancer biology. The main aim of the program is to discuss the cutting edge of technologies that have made significant contribution in the field of biotechnology in enabling the development of and for example, a better healthcare, the novel molecules in new drugs, advanced diagnostic tools, enhanced food security, improved supply of potable water, more efficient industrial development processes for transforming raw material that support UN SDGs and offer new opportunities for global partnership. I trust that two-day conference will be the platform of many debates and discussion and the speech of eminent panelists with enlighten the participant to do more for the sustainable development and human welfare. My best wishes to the conference team headed by Professor Amjad Abdin Saab and also all the panelists and participants in the conference. I wish the conference with all success. Jai Hind. Thank you.
Thank you, sir. Thank you for the informative message and constantly inspiring us to do good for the society and for the institute. Now, as respected registrar sir could not join us today because of some urgent work, so I would request the convener of the program, Dr. Javed Ahmed Sheikh, to please come up on the stage for the vote of thanks.
All the dignitaries on and off the dais, respected faculties, eminent speakers, and dear students. A very warm welcome to all of you. So it's an absolute honor to have been asked to offer a vote of thanks on this occasion of this National Conference on Biotechnology. And on behalf of the entire organization team, we heartily express our gratitude to all the dignitaries, distinguished guests, and chief guests for adorning the dais and gracing the occasion with their solemn presence. It's a great honor to host all the members and audience for this August meeting. I'm thankful to all the participants for coming here and attending the inaugural session and marking this early morning start for this two-day conglomeration. So Jamia Hamdard's commitment towards this highest quality education and world-class research is indeed a reflection of the great leadership that we have. Though we are very unfortunate that Honorable Chancellor Janab Hamad Ahmad Sahab could not join us for the moment, but being Chief President of this national conference, that's the testimony to the great support and blessing from our benefactor, that's Hamdard National Foundation, that they bestow upon us, and we will be always indebted for that. We have been very fortunate, and we have a renowned identity like Professor Asim Ali Khan Sahab, the Director General of CCRUM, who imparted such sagacious insights. His presence has been a great encouragement and source of motivation for us. The organization headed by his good self is surely a golden example of human welfare, wherein they are relentlessly working towards the human health through sustainable methods. So your valuable contribution towards healthcare is worth emulating for all of us. We thank you. And then leading for the front and guiding us at every step is our Vice Chancellor, Honorable Professor Dr. Farah Alam Sahab. So whom we profusely thank for this unflinching support and guidance throughout the program. His dynamism and overwhelming interest in the activities like these have helped Jamia Hamdard to inch forward towards competitive world is worth mentioning and appreciating. Thanks a lot, sir, for your continuous support. I would also like to express our gratitude to our Dean, Professor Raisuddin Sahab, who has been generous enough to increase the dice. His scientific inputs and unmatched support towards fulfilling the objective of this conference has been enormous. We can never thank him enough. And also like to express our gratitude, though in absentia, towards our Registrar Sahab, Mr. Said Saud Akhtar Sahab, for his immeasurable cooperation at every step of conceiving and executing this conference. And all the deans that are present here, the HODs and the teaching staff, various schools and the distinguished participants, we offer you a very gratitude for their active participation, and special thanks to our students who are a part of this distinguished event today. Thanks to Imam Sahab for reciting these beautiful and relevant verses, and the University Tarana team for infusing the vigor through melody. Last but not the least, thanks to the conference chair, the organizing secretary, Professor Abdeen Sahab. Is indeed a pleasant duty I take upon myself, and working with him has been an absolute pleasure. And with greatest of my gratitude, I acknowledge the amazing leadership which he elicits and continues to be instrumental in giving life to the events like these. Thanks a lot, sir. Finally, I am thankful to the entire participants as well as sponsors of this conference. Not only that, I must express the gratitude and profound sense of gratefulness to all the guests who are with us today for these 2 days. And so let us celebrate the event, learn and unlearn, and keep moving forward, hand-holding each other and remaining committed to the ethos of this great school of learning, working for the human welfare with the guiding dictum of being Hamdard. Thank you. Thank you all. Have a blessed day.
Thank you so much, sir. Now I request our Honorable Vice Chancellor, sir, to please honor the Honorable Chief Guest, Professor Asim Ali Khan, sir, with a shawl and moment. Next, I would invite respected Professor Raisuddin, sir, to please felicitate our patron of the program, Honorable Vice Chancellor, sir. Now, I would request Professor M.Z. Abdeen, sir, to please present the memento to the chairperson of the event, Professor S. Raisuddin, sir. Oh, yeah. Next, I would request Professor Raisuddin, sir, to please present a token of appreciation to Professor M.Z. Abdeen, sir. Thank you, sir. Now I request everyone to please stand rise for the national anthem.
जन गण मन अधिनायक जय हे भारत भाग्य विधाता पंजाब सिंध गुजरात मराठा द्राविड़ उत्कल बंग विंध्य हिमाचल यमुना गंगा उच्छल जलधि तरंग तब शुभ नामे जागे तब शुभ आशीष मांगे गाहे तब जय गाथा जन गण मंगलदायक जय हे भारत भाग्य विधाता जय हे जय हे जय हे जय 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 हे
Thank you so much. Now it's time for the tea break. I would request everyone to please move towards the lobby area. The tea is waiting for you outside. I request all the participants to please go to the registration counter and collect your kits. I repeat, I request all the participants to please go to the registration counter and collect your kits.
मैं बोलता हूँ हेलो 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 कितना कर दिया हेलो कर दिया तो जाएगा आवाज आ रही है ना यहाँ पे आवाज यूट्यूब पे आवाज नहीं आ रही क्यों तेरी आवाज नहीं आ रही बात अभी तो आ रही है हेलो हेलो इसमें एक फोल्डर होगा उसको डेस्कटॉप पे ले लो कैसे ले जाएगा सर पे फोल्डर नहीं है सीधे ये हाँ वही वाले जो फाइलें वाले वाली सेटिंग आई है तो जगह देख लो आवाज की नहीं आ रही सुनना भाई फोटो ले ले एक तो फोटो ले ले बढ़िया एकदम खूबसूरत करना जो आएंगे दिखा कर चलेंगे सर पेन ड्राइव
ये नाम से जिससे कि उससे सेव हो गई है ना
हाँ इसमें भी है नहीं ये हमने उसके लिए रखा है बैकअप जूम मत कर भाई
यार ये आवाज यार कैसे नहीं जाएगा
अभी आ रही है बोलते हैं वही मेरा लेना कर नहीं बैठ जाए चीफ गेस्ट की जगह नहीं जब लाइब्रेरी माइक माइक कर दिया आप सब रिकॉर्ड करेंगे सारी बातें खुल जाएगी
बातें जा रही थी
हाँ,
स्क्रीनिंग में
अभी नहीं जा रही थी अब जाएगी फॉलो माइक चाहिए आपको
दिखाएंगे तब
लगाना है
चेयर को वेलकम करेंगे
वो मैं कर हाँ वो सब खुला करना है हाँ हाँ करना है दीपक निकल
बाहर जो भी हमको बोलो अंदर
खुलवा लीजिए आ जाओ
Welcome back. Now we are entering in the first plenary session of today, and now is the time to hear out from some great visionary and thought leaders in the field. To chair the session, I consider it a great honor to welcome Dr. Ajit Kumar Shasni on dais. Dr. Ajit Kumar Shasni is the Director of National Institute for Plant Biotechnology. Dr. Shasni did his PhD in molecular biology and biotechnology from IERI New Delhi and postdoctorate from Purdue University, USA. Dr. Shasni has published various research papers of national and international repute and around 101 patents in his name. He has various awards to his credit, such as Samantha Chandrasekhar Award, CSI Technology Prize for Biological Sciences and Technology, and many more. He is the person behind complete genome and transcriptome sequencing of Holy Basil. We are honored to have you here with us, sir. Now, to co-chair the session, I would like to request Dr. Reema Pant ma'am to please come upon the dais. Dr. Reema Pant is an educator and environmentalist, a teacher by choice and a lifelong leader. Dr. Pant has over 20 plus years of research, teaching and working experience in different academic institutions of national and international repute. She is presently associated with Graphic Era Hill University, Dehradun as a Director of Center for Idea, Innovation and Environment. She taught Welham Boys and Welham Girls School for college for close to 16 years. As a practitioner of hands-on teaching, learning, she has started an organization, Techno Hub Laboratories, that work in the domains of education, technology, and environment. We welcome you, ma'am. And now is the time for the keynote address, which will be delivered by the man behind the genetically modified mustard, which is India's first transgenic food crop to be granted approval, which has just happened last month. So please, a big round of applause to Professor Dept.of sir and his team. This is really a matter of great pride and honor to all of us. Sir, indeed need no introduction, but for the young generations, please allow me to say a few words. Sir is a professor of genetics and former vice chancellor of the University of Delhi. He is a noted researcher whose current research interests lie in the development of transgenics and marker-assisted breeding of crops. A new frog species, Minervaria pentale, discovered in Western Ghats by a team of Delhi University researchers, was named after Professor Pentel. He has many research papers and awards to his credit. To name a few, he was awarded Officer des Palmes Academics for contributions to research and education by the Government of Republic of France. He also has received Lifetime Achievement Award by Mahindra Samriddhi Foundation. He is the member of National Academy of Agricultural Sciences, National Academy of Sciences India, Indian Academy of Sciences, and Indian National Science Academy. Now I request Sir to please enlighten us with his keynote address on the past and future of agriculture.
Get the slides. This is. Yeah. This is to go forward and back. And this is pointed. Good afternoon. I was going to say good morning, then I checked my watch to see whether it's still 12 or it's gone beyond 12. I'm very happy to see lots of students here. You are the future of science and technology in this country, and therefore I'm delighted that so many of you are here. Thank you for the invite to be here this morning. And the topic I have chosen is a very big canvas topic. And I don't know if I can do justice to the topic in 30 minutes, but I'll try to share some thoughts with you that, on this issue of food and agriculture and self-sufficiency in food. Homo sapiens about 200,000 years back, and then they spread out of Africa into Asia and Europe. This is just to remind us that study of evolution is the key to understand biology and key to understand even some of the modern days dilemmas. Why I'm showing this slide is because evolution is the theme behind all studies in biology, but also to make the background for the next slide. When human beings reached Europe, for example, it was a frozen mass. There was an ice age going on, and only until then, the structure of human populations was tribal in nature. Bands of tribals in Africa which moved out, and tribal structure in human evolution. Agriculture only started about 12,000 years back. I don't know if this is working. About 12,000 years back,
globe warmed up, and that is when agriculture started. Otherwise, human societies were all tribal in nature. We still see tribalism in human, in all our modern societies also, but that is for some other day. We can discuss that. This is about agriculture, and I want to emphasize to you that agriculture started when global temperatures came down, ice melted, and the rivers were flooded with water. That's how the agriculture started in the, let's say, in the Fertile Crescent in the Iraq, Turkey area, in Euphrates and Tigris rivers. This is the great Vavilov's centers of origin of agriculture. His hypothesis was that centers of diversity of a crop, both in terms of the crop species and its wild relatives, is the center for origin of that. And this is the most important area here, the Fertile Crescent, where domestication of plants and animals took place first, about 12,000 years back, where wheat, barley, alfalfa, many other crops and many animals were domesticated. This is where the sedentary mode of life started. Then, of course, in China, in other parts of the world, in Central Americas, and in South America, and so on and so forth. So agriculture started the sedentary mode of life, and all modern political structures which we see are due to how to keep law and order, history. In 1900 and up to and this, in 1900 the average life expectancy in the Indian subcontinent and all of us in the old age are living off the benefits of modern medicine. Dr. Brati will be talking about
To reach the other end, where it can benefit patient, it has to cross this bridge, and we call it crossing this bridge, crossing the valley of death. That means a lot of these ideas that are created or generated in the lab never reach the other end to benefit the patient, and that is because a lot of them die in this long journey of crossing this valley of death. Now, in order to make this easier, in last 10 years or so, a new, new science is emerging, which we call translational science. And in a crudest sense, this is turning basic research into medicines and treatments. Now, why this has not been possible more frequently than is likely to happen in coming years? Because this translational research, number one, is highly complex, requires expertise of various kind, is very expensive, requires a lot of patience, lot of time, and lot of money. I am giving you an example from a paper that was published in American Journal of Medicine. And there, the authors have reviewed research between 1979 and 1983, where they identified that there were 101 discoveries that were reported, and the claims were made that these will result into some treatment or some therapy for improving the quality of life for human beings. But by 2002, only 5 had received license for clinical use. So out of 101, in almost 20 years, only 5 received license for clinical use. That means the chance of success are very, very low. That is about 5%. But even when that happens, it takes very long time. In this journal report, we could see that from the patent or publication of your discovery to a product, it takes anywhere between 14 to 44 years, the median being 24 years. So it's a long journey, requires a lot of money, a lot of time, a lot of patience. Vaccine development timeline is also very long, and I think thanks to corona epidemic and the subsequent vaccine development, lot of this has come into public domain. A lot of people have read about it. We do research in the lab, identify, let's say, vaccine candidate. We carry out preclinical testing in the animals, which takes anywhere between 4 to 5 years. Once you have demonstrated preclinical efficacy and safety in animals, you are given regulatory permission to test in humans. You go through various phases that are phase 1, phase 2, and phase 3. Phase 1 is actually a safety study in humans, where you are allowed to test it in a small number of individuals, let's say about 100. If it is safe, then you are allowed to go for phase 2, where you are allowed to test more than 1 dose of vaccine. On the safety and dosing studies, if everything goes all right, you are allowed to go for phase 3. Phase 3 is efficacy study, where you have to demonstrate that your vaccine is not only immunogenic, but also is reducing the incidence of the disease against which this particular vaccine is being formulated. Now, this is done in several thousand individuals, and it's a multicentric study. With demonstrating the safety and efficacy after phase 3. If you are lucky, you can get the regulatory permission to market the vaccine in the market. Okay, but the story doesn't end there. You continue to do for next several years the phase 4 study, that is the long-term safety study. And if there is a problem in phase 4, you have to come back to lab and start all over again. So the whole process takes anywhere between 15 to 20 years. Now you may ask, these corona vaccines were allowed to be made in 6 months to about a year. But every time, if you remember very carefully, all of these vaccines were allowed to be used as emergency use authorization. Okay? There was a real emergency, and there was no way that we could have waited for 15 to 20 years. So emergency use was made possible for these vaccines. So development of a vaccine is long, about 15 years it takes. It's risky, only few ideas become products. It's a complex, requires multiple expertise, team of experts, requires a lot of coordination, a lot of money, a lot of patience. As I said, translational research for vaccine is a team game. You need scientists, you need biotechnologists, you need product development specialists, you need clinicians, you need statisticians, you need public health specialists, you need population biologists, and you need social scientists. Now I have, provided pictures of some of the key, leaders of this team. Dr. Bhan, who was a professor at All India Institute of Medical Sciences, and later he became Secretary to the Department of Biotechnology, was the person who made an observation in his clinic. A similar observation was made by Dr. Durga Rao at IISc Bangalore, led to identification of virus, and I and Harry Greenberg, being virologists on the team, we developed all the assays that are required to develop the vaccine, to test its quality, and then to test its efficacy in the field. These are 4 clinicians: Neeta Bhandari, Gagandeep Kang, Roger Glass, and Ashish Bowdekar. These are in 4 different locations. They carried out the clinical testing of the vaccine in field and in hospital. And all the clinical material coming from these clinical centers was coming to our lab, which was a GLP qualified lab for testing the vaccine efficacy. So every time, over 2.5 to 3 years, we tested several thousand stool samples from kids looking for presence of rotavirus. We were testing the immune responses in these kids, looking for efficacy of the vaccine. A little bit about rotavirus. This was discovered in 1973 by an Australian scientist, Ruth Bishop. This is a non-enveloped triple-layered virus. The important is that there are 5 serogroups of rotavirus: A, B, C, D, and E. And humans are infected primarily by A, B, and C. All of these 5 serogroups cause disease in animals also. Now the genome is, comprising of 11 double-stranded RNA pieces. So that means that, if in animals or in humans, an animal virus and a human virus infect simultaneously, there is a possibility of reassorting these genes, resulting in generation of a reassorted virus, that is human-animal virus or animal-human virus. And this virus could have, varying pathogenicity. It may have increased pathogenicity, it may have decreased pathogenicity, or no pathogenicity. And new serotypes or new genotypes of virus continue to emerge. So there are 2 important proteins of this virus, VP4 and VP7. These are outer capsid proteins. A VP4 is involved in virus entry. Please remember, VP4 is involved in virus entry, so it must be interacting with the receptor on the human cells. And VP7 is immunogenic glycoprotein, which generates antibodies which are highly immunogenic. So there are several gene sequences for VP7, and there are 16 of them known so far. And these are designated as G-type, G1, G2, and G12. And there are several VP4 gene sequences. There are 28 of them so far, and they are designated as P-type, P4, P6, P8, P9. So a combination of G and P generates or defines the genotype of a rotavirus. And if you look at the distribution of various genotypes worldwide, you find the G1P8, the virus containing the BP7 of G1 type and BP4 of P8 type. They constitute almost 50% of the virus genotypes, and there are several others at different frequency. Now, this virus is transmitted through fecal-oral route. Virus enters the gut. It binds to the cells of the intestine. It invades, these cells and starts destroying these cells. And this leads to malabsorption of carbohydrates. This results in diarrhea. So, virus is actually responsible for diarrhea, and more so in children. Diarrhea is caused by many viruses and several bacteria, but rotavirus is the leading cause of severe gastroenteritis in children, specifically less than 5 years of age. Almost 30% of all severe acute gastroenteritis cases across the world are caused due to rotavirus. Now, this, the global map is showing number of deaths due to rotavirus diarrhea. Each spot is about 2,000 deaths a year. Okay, and you can see India, there's absolutely no space left. So there are a huge number of rotaviral deaths that take place in India. Rotavirus causes diarrhea, it causes vomiting, and in severe conditions, it can cause dehydration, shock, and death. As I said, rotavirus accounts for approximately 1/3 of nearly 600,000 global child deaths, which are attributed to diarrhea. 95% of these deaths are occurring in developing countries. You can see, in US or in Europe, they are almost, or in Australia, there are hardly any rotavirus associated deaths. What was the scenario in India before the vaccine got introduced? There are 25 billion children that are born each year, 20 million episodes of rotaviral diarrhea by 5 years of age, 400,000 hospitalization, and about 100,000 deaths each year. That means one death due to rotaviral diarrhea for every 250 children born. So this has been a serious problem for this country. Now, this is a virus, this is an RNA virus, as I said, and there is no antibiotic to these viruses. And what is the best bet is the vaccine. And this has been time and again proven. This is some data that shows introduction of vaccine in an African country, where these are the number of rotaviral diarrhea cases before the vaccine got introduced, and subsequent to vaccine introduction, you can see this number of cases are plummeting. Okay, so the first vaccine was made in the US in 1998, and this was a live attenuated vaccine using the Jennerian approach. If you remember, Jenner made the vaccine where he used a virus which is similar to the pathogen that was causing the disease. So rotavirus was taken, which was not causing any clinical symptoms, or was less pathogenic or non-pathogenic, and this vaccine was marketed by Wyeth, and the name of the vaccine, or the commercial name, was RotaShield. This was a human simian rotavirus reassortant. I told you, these reassortants have different biological properties, and they are very useful for making vaccines. Okay, so this vaccine had undergone phase 1, phase 2, phase 3 trial, and was implemented, was allowed to be used in field. But after 1 year of follow-up, after phase 3 and phase 4, it was found that vaccine was associated with intussusception. Intussusception is actually telescopic folding of a small intestine that can cause edema, necrosis, and perforation. Even though the frequency of intussusception was very less, 1 in 10,000, but vaccine is meant for healthy children, and even one death in 10,000 is unacceptable. And therefore, the vaccine had to be withdrawn even after going through all 3 phases of development. So I have discussed this, and so in phase 4, this vaccine had to be withdrawn. Then in 2006, 2 new vaccines were described. One was Rotarex by GSK. This was G1P8 virus, and another was Rotatec by Merck. This was a pentavalent vaccine using 5 different viruses. This time, vaccine was tested in 16 to 17,000 infants. And I told you the frequency of intussusception was 1 in 10,000. So if there was intussusception associated with any of these vaccines, we would see 6 to 7 cases. And thankfully, no such case was seen. There was no increased risk of intussusception, or there were no serious adverse events. And efficacy of these vaccines was very high, somewhere 95% or 98% against severe disease. Now, based on the efficacy of these vaccines, in 2009, WHO made a global recommendation of use of these vaccines in infants. Now, where was the problem? These vaccines were highly efficacious, 98% against severe rotavirus disease, so they were very, very good, but they were very expensive, $250 for 2-dose or 3-dose regimen. Now, how many people in a country like India or any other low-resource country would be able to afford this vaccine? And therefore, we needed a vaccine that would be affordable, at least in low and middle income countries. Other than this, another problem that was encountered was the following. When this vaccine was used in Ghana or Bangladesh or in some other South African country, the efficacy dropped to around 50 to 60%. Now, this was not something that was not known to us. We have seen in this country that oral vaccines have low efficacy in this country or in low and middle income countries. Now, why do we think oral vaccines have lower efficacy? There are several hypotheses, but we just thought that in these countries, breastfeeding is something which is very common. And as soon as you give any vaccine to an infant, they start crying, and mothers have a tendency to start feeding them. And we know that mother's milk at that stage contains high amount of antibodies, and a significant proportion of these antibodies are against rotavirus. So we hypothesize that it is possible that mothers feeding these children is leading to neutralization of virus and therefore low efficacy of the vaccine. And the second issue was obviously the affordability of the vaccine. Nobody could afford $250 vaccine. So we just hypothesize that if a vaccine is made from an Indian virus that establishes well under the conditions of breastfeeding, can we make an effective vaccine for India? Can we make a cheaper vaccine for India? And this is where the story started from the All India Institute of Medical Sciences in 1985. Okay, you will see the long story. So Dr. Bhan was running this clinic of infants at AIMS, and he noticed that so they were doing a rotavirus surveillance. So randomly kids were being checked for rotavirus presence, and he found that almost 50% of the infants were getting infected if they had lived in the pediatric ward for about 3 years, about 3 days, and 75% of them were carrying rotavirus if they had been there for about a week. The virus was isolated and identified as virus G9P11. This was a human bovine reassorted. This was a virus which had never been described. So it's a novel virus. Okay. And these kids were showing no symptom of diarrhea. Okay. So it was then reasoned that maybe this would be a good vaccine candidate if we could develop this as a vaccine. So he followed up these kids for next 2 years and wanted to see whether there was a vaccine effect seen. That means that were these kids showing any resistance to development of diarrhea over the next 2 years, and they did. Okay, so these observations made us believe that maybe we had a vaccine candidate that could be developed clinically to produce a vaccine. Similar observation was made in Bangalore. But there was a different virus over here. It was called I321. It was a bovine human reassorted. This was human bovine reassorted. This was G10P11. So a different virus, but similar observations were made. So as I said that, one virus, this is 1160 virus, where majority of the genes were human, except one gene, VP4. You remember I said VP4 is the protein that is involved in interacting with the human cells. And then we had another virus, I321. Majority of the genes were of bovine origin, except VP4 and except NSP1 and NSP3. But VP4 again was of animal origin. So we thought that maybe this virus is not interacting very well with the cells, and therefore this is causing less of pathogenesis. So with all this background, we thought that these two viruses had the vaccine potential and must be developed as vaccine to see whether they had the vaccine effect. So I will not go into details of the various steps that we took. The virus had to be adapted into several cell types, and finally, the virus was grown in African green monkey kidney cells by an American company at that point of time, 1996. Bharat Biotech did not exist. There was, except Serum Institute, there was no vaccine manufacturing company, and even Serum had no experience of making a novel vaccine. They were just producing the vaccine that had been produced for years after years. So the vaccine was produced by Flow Laboratories, and it was tested in, and there was no experience of clinical development of any vaccine. So vaccine was tested at Cincinnati Children's Hospital in USA. Now the vaccine is meant for infants, but nobody is going to allow you to test the vaccine in infants. So the vaccine is first tested in adults, then in younger children, older children, then in younger children, and finally in the infant. So it was done in the US, and what we found that both vaccines were attenuated and safe. They replicated poorly in adults and children with pre-existing rotavirus immunity. And we found that 116C replicated somewhat more efficiently and was more immunogenic than I321. Now, this vaccine was produced in African green monkey kidney cells, and at this stage, we moved all our studies to All India Institute of Medical Sciences in New Delhi. Again, the studies were conducted in Indian adults and Indian children, and we found similar results that were found in the US. So we carried out immunogenicity and safety studies in Indian infants at All India Institute of Medical Sciences. Vaccine was still produced by Dynacorp in USA. And what we found that 116C was more immunogenic than I321, and 116E was shared in the stool more frequently than the I321. So, these studies demonstrated to us that both 116C and I321 both were safe and well tolerated. 116C induced significantly better immune response. 116C replicated well in infant gut, indicating that the vaccine take was better for 116C than I321. At this stage, we chose to work only on one vaccine because two vaccines was very expensive. And we had to start using the Vero cells for making the vaccine, because around this time, WHO recommended Vero cells as the substrate for making human vaccines. So the vaccine was adapted to grow in Vero cells at Bharat Biotech. This is when Bharat Biotech got involved, and we made a Vero cell vaccine. Now, since the vaccine had been made in Vero cells, there was a different substrate. We had to start all our studies in clinical development from phase 1, phase 2 again. So in the Vero cell derived vaccine was again demonstrated to be safe in younger children and then in infants, and we found that 116C we were allowed to test 2 doses of vaccine, 10 power 5 and 10 power 4. And we found that 10 power 4, that was higher dose of vaccine, was more immunogenic than 10 power 4. Okay. Now, demonstrating the safety and immunogenicity does not mean that the vaccine is efficacious. So, vaccine efficacy had to be tested in a phase 3 trial in large population, and we chose to conduct these studies in 3 sites in Delhi, Pune, and Bangalore in 6,800 children. First, we demonstrated the the incidence of rotaviral diarrhea in this population, and then the population was given vaccine, and subsequently we followed them up for 3 to 4 years to see whether we were able to reduce the incidence of rotaviral diarrhea. In 2014, we published the results of phase 3 trial, and what we found, the vaccine was efficacious, it was safe. However, the efficacy was 55%, which was very similar to Merck and GSK vaccine. Now, this was a bit of a setback, but nevertheless, it was still efficacious and still a usable vaccine. Now, this vaccine in 2014 was launched into the Indian market by Prime Minister of India. In 2018, the vaccine received WHO pre-qualification. That means the vaccine was used in India and could be used in countries outside India. In 2019, the vaccine received permission to launch in India's universal immunization program. As you can see, now vaccine, besides several other vaccines, rotavirus vaccine is part of the universal immunization program in India. Now, This is a journey of 30 years, and I had the privilege of joining this journey, a significant part of this journey. In 85, we started with the clinical observation. 88, virus was isolated in the lab that we call bench, and then clinical development. In 2014, the vaccine was allowed to be marketed, and 2016, the vaccine is available in the Universal Immunization Program. A large number of individuals were involved in making this vaccine, and I have listed the acknowledgement section of the Lancet paper over there. But what is important is that here we have a vaccine which is made from Indian virus, researched in India, made in India for Indian kids as well as for kids across the world. Now the vaccine is being used in several low and middle income countries besides India. Through this journey, I also traveled. I started my work with rotavirus at National Institute of Immunology and moved to Translational Health Science and Technology Institute, where I carried out the phase 3 trial. Now, stop over there. Thank you very much.
Thank you so much, sir, for sharing your work with us. We appreciate your efforts in making of the Indian rotavirus vaccine. This will definitely help the mankind. I request the audience, if they have anything to discuss or if there are any question, they can ask. Mike? Okay.
Can you please speak up? I can't hear.
Number one, how long it protects? Number two, can you use this for tracking?
The first question is clear. Okay. So, the vaccine will, even if you don't take vaccine, by 5 years of age, everybody is immunized by natural infection. Okay. So, and actually, you don't need vaccine in young children. It is actually required for infants because that is where they are at risk of losing life because of severe diarrhea. So this is perfect for protecting infants. What was the second question? Okay, not this vaccine, but I had a discussion with ICR people, and they would like identifying a non-pathogenic rotavirus, but not this vaccine, perhaps no.
Thank you so much, sir. The next distinguished to honor us with his presence is the Professor Sumanpreet Singh Khanuja, sir. He is the founder and chairman of Flora Fauna Science Foundation, Lucknow, and a distinguished professor of bioengineering and food technology in Shulini University, Solan. He is the former director of CSIR CMAP. Dr. Khanuja is also the founder and mentor of Skies India Innovation Incubator and Food Innovations Venture. He has published more than 200 research papers, 82 patents, and 7 books to his credit. He is the steering person behind various missions such as Leher for farmers, Tarang for enabling students, Sakshma for enabling women entrepreneurship, Manav for reaching the unreached part of the society, NutraSky for nutraceutical industries aiming food for health, and many, many more. With the long list of his achievements and glory, I would like to invite upon the stage Professor S.P.S. Khanuja, sir, to please put light on the topic of eco-agriculture.
Thank you. I thank my friend Abdul here, who invited me before COVID and now post-COVID. Not in a sense post-COVID, but overcoming COVID. I had interacted with you last time on some of the things that concern with agriculture. Now, after two elegant talks, one on past and future of agriculture and other on health sector for rotavirus vaccine. I would like to take you into the present and translational part of agriculture. My belief is that we might keep doing many things in laboratories, but unless and until they reach the farmers and entrepreneurs and it changes their scenario, it will become an open-ended game only. So that is the reason that I had taken voluntary retirement from CSIR when I was 49 and tried to establish a foundation where I could ask the institution like mine to come to the field with me and tell this farmer, or a villager or the sons and daughters of the farmers that how they are going to make a difference in their life with the technologies or science that you are developing. And also it becomes important that if you have done some science, you believe it's a breakthrough, then if there are shortcomings when it is translated, it goes back to the lab and comes out with all those bottlenecks covered and something that really makes people happy. And that's why my dream for India is Prasanna Bharat. That means happy India, and happiness comes through eco. When I say eco, it is economics and ecology. So unless and until these two come in balance, no one is going to take care of any of those big words that we keep talking about, either the germplasm or diversity, biodiversity, the biowealth and so on, because, ultimately a man sitting in a village or even in urban areas without an employment won't do anything that ethically is very good unless and until it is economically also good. And with the concerns that are coming up today with environmental calamities that you are seeing, whether it is biological, physical, or chemical, we also have to think of ecologically viable technologies. So I'd like to take you through a sort of journey where I'll tell you that how, rather than talking of cultivating food in fields or crops, the farmer could be doing cultivation of health in the fields, and through that also earning much, much more than what otherwise one would earn through typical traditional food crops. I don't believe necessarily that nothing should be imported. We should be self-sufficient. That is what is the meaning of Atmanirbharta. I believe that you can import if it is economically cheaper and export what is economically expensive. And if you do that, then rest of the things for healthcare and others you would be able to do. So that's what I would try to do. I have 25 minutes, sir. Okay. Okay. I would like you to take a glimpse on Norman Borlaug, the man behind Green Revolution. Dr. Pental also talked about him. But I am going to talk about him from a different perspective, not only Green Revolution that he brought by dwarfing genes in wheat and making multi-folds yields in wheat. I had a fortune of meeting him in my own field when I was doing wheat breeding at Pantnagar in early 80s or late 70s. So at that time he was visiting India and watching, what is happening with wheat that really has brought the revolution. And when I interacted, I found a mentor in him. So he talked a lot, and for me it was a fascination because I was MSc student and a Nobel laureate talking to you. Informally it becomes really motivating, and that's what made me to decide that I'm going to remain in science, whether I do basic science or applied science, or go out of science and translate that science. So he gave some words which I have assembled here. Plants do not speak, they just whisper. And therefore, you have to be real close to them to understand that. Unless and until you are close to the plants, you cannot understand them. Reason? Animals still, they can bark or mew or roar. And human beings, they do all sorts of nonsense with their tongue. but the plants, they don't speak out. And when they don't speak out, we have to understand what are they saying. And that communication, once we start learning, then we can do wonders in agriculture, plant science, environment, and economics. So we have to break those limits. So what I suggested long back to Planning Commission also, and it is getting into now practice, is I3 model. That is inclusive, integrative, and innovative. We have to think beyond, we cannot say, like in science, what is science? Science is answer to two questions. What? What is it? If you can explain it, and why is it? Then you are into science, business of science. And as soon as you shift into how, the answer to how is it happening, or how that doesn't happen, it becomes technical know-how, and therefore that is technology. The science becomes technology when what and why become how. And when you do how not and why not, when you answer why not, you are an innovator. When you say how not, you are an entrepreneur. So that's what the model has to be there. And clubbing these, then we can say that we perhaps can cultivate health in farmers' fields. And a farmer can be, in my opinion and my vision, a farmer should say, I am a protein farmer. I am a carbohydrate fiber or soluble fiber farmer. I am omega-3 fatty acid maker farmer. Like that. If farmer would start understanding only that much aspect, that what is he doing, like my farmers could understand that they are making anti-malarial drug in Artemisia annua. The economics from them changes. Industry has to understand and be at same level to talk to them, and therefore they will gain more. So looking at that angle, and this was a report Ajit had shared with me long back. It's almost beginning of this millennium, which CII and McKinsey had given to us. I interpreted it in a different way, that we start calling ourselves India as developed nation sometimes or developing nation sometimes. What are we? If we talk in terms of the lifestyle disorders that we suffer, we are more than developed, because all those lifestyle diseases that occur in the wealthiest world, developed world, are here. And therefore, while it is a threat, it is also an opportunity. If we can work in these areas, we can develop drugs and molecules or natural products that can take care of it. We are sitting on $1 billion or multi-billion dollar situation, like hypertension or nervous system, diabetes, and so on. And I was talking about the whisper of the plants, which I learned from Dr. Norman Borlaug. I understood that plants, they also communicate. And that communication is how? Like we say, there is a choreographer and there is a dancer. And you must have seen so many reality programs coming, like Nach Baliye and so on, Dance India Dance. So whenever they perform good, they say there is a very good chemistry between the choreographer and the dancer. So dancer here is scientist and choreographer is plant. So if you can have a good chemistry, that means, do you understand chemistry of plants? You can do biology there. And if you do that biology through a chemistry, real agriculture in economic terms will happen. And that's what we understood when I took the leadership of CMAP, Central Institute of Medicinal and Aromatic Plants, that this is a chemistry which is important. This is how plants communicate. This is their secondary metabolism. And selfishly, human beings call it medicine for us or drugs for us. And that's how medicinal plants are medicinal plants. And if we really start translating them, we have primary metabolism, what you are seeing without the boxes, and the gray boxes shall tell you the secondary metabolites, which accumulate there. And if you see primary metabolism, I have written food, functional foods, nutraceuticals, and food ingredients. And in secondary metabolism, you get aromas, you get nutraceuticals, you get food ingredients, and you get pharmaceuticals. So this is the wealth that you can derive out of plants. And therefore, we can talk about natural products in a modern biology sense. I'm not talking of Ayurveda or Unani or Siddha or homeopathy only. They are important. But if we are talking of modern biology, which validates the use of these molecules as drugs or the molecules contained in the plant materials that you are going to use with a mode of action defined, safety and toxicity well defined, then you are sitting on a big industrial platform. And recently when we had COVID, we were talking about this, comorbidities, immunity enhancements, and we started talking about Kara and other things. But I would say, well, that was a time when you took anything which was told to you that is good for you. But if we are talking in terms of the natural product which are going to give you that help, then this could take care of stresses, metabolic status, age-related things. Aging population has to age healthily. They should have good energies. They should be feeling happy and other undefined factors. So that means we can change the spelling of farming from F-A-R-M-I-N-G into P-H-A-R-M-I-N-G. And if farmer starts doing that, then it's cultivation of health. So I'll give you some example where you could be managing your health, you could be preventing diseases, and agriculture will be the tool as well as a resource for that. And the first example I like to give you is of obesity. Now, you know, obesity is not as such a single disease. It is consequence of many factors and the precursor for many diseases. And obesity doesn't mean that a man looks fat necessarily. If you look at the chemistry of the blood or chemistry of the metabolism, then you find that a person may be obese because of whether you are talking in the cholesterol or cardiovascular health and so on. So it could lead to cardiovascular problems, nervous problems, stroke, and bone-related osteoarthritis, sleep problems, and cancer. So it's a gambit of diseases that would ultimately come up if you are obese. And can we manage obesity through molecules which plants produce? Yes. I've taken some examples. It's a huge list, and many of you at Jamia also worked on those plants. So simple example, red apple, pyruvate, that's right, an apple a day keeps the doctor away. Your tea, which has flavonoids or catechins, your Garcinia cambogia, which produces hydroxycitric acid, which can literally reduce your weight because of the cholesterol management, and allium, sativum or garlic or lessons. The largest selling raw herb in the globe, in the world, is allium. If you go to Europe, those people who eat a lot of chocolate, because of chocolate, they eat a lot of sugar and fat, can also do eat a lot of allium and keep themselves in balance, whether you talk of Germany, France, and so many other European countries. Diabetes, you know that now, literally, if anyone says I am a diabetic, you don't get surprised. Earlier you used to say, oh, aapko sugar hai. Now you don't say that. You know, you say, oh, he has. So everyone who comes to you and you ask whether you will like to take sugar in tea or not, because it's not a surprising factor. So that means the frequency of happening of diabetes is too much now. And you have answers also. For example, bitter gourd, it has a protein, that is P-insulin, which can help you in maintaining the insulin levels, and also has secondary metabolites which can really do wonders in case of curing of the diabetes. For ivy gourd, coccinia, there also you have secondary metabolites like your bitter gourd, and trigonella or methi. It has a twin system of helping you in diabetes. One is physical, that is, if you just soak these seeds, you get a gel, that is a soluble fiber, which absorbs the fat and sugar from your diet. So if you are eating chocolate, if you have this also, then that is going to make a gel of the fat and sugar and excrete it. So दावत के टाइम पे अगर आप ये भी खा लेते हो, तो you are going to save yourself. And then stevia, which also can help you not eating sugar, but have the sweetness 100 times more than what sugar has. And when you have stevia and sugar in a synergistic combination, it is 250 times sweeter. So that's what one could be thinking. Then brain health is also an important issue. And you have plants for that. You have Bacopa monieri or Brahmi. You have Centella asiatica or Mandukuparni. You have Acorus or Vach. You have Nilambo or Kamal. So they have those molecules which help you in memory enhancement, they help you in your cognitive response, they help you in many, many other ways when child is developing the brain. So that was pharma part of it. But let us think beyond food and pharma. Now I talk of nutrition. And nutrition, the answer lies in functional foods. What is a functional food? It's typically a food. It doesn't have to look different, nothing. It should look like feed, chapati, idli, dosa, whatever you eat. That should have some added advantage because of its metabolic ingredients of the metabolic ingredients of the components of that food, which give you the advantage beyond normal hunger fulfilling. And that's when it starts helping you medically. You call it functional food. And functional food is functional because of the molecules, those are called nutraceuticals are present in them. Nutraceuticals are those, if you extract and purify the molecules which are really doing that medical help to you or pharmaceutical help to you, then that is the reason. So rather than going to nutraceutical and making into capsules and pills, you could be taking advantage of nutraceutical in understanding the mechanism and dosage and toxicity or safety, and make functional foods which public will like to eat. And functional food growing for farmer is going to be a very big, wonderful eco option. You know that in biotechnology, this work, golden rice, where you talked about beta carotene synthesis in golden rice from a normal rice, and the genes were incorporated from daffodil and a soil bacterium, Erwinia, and you created a typical pathway which was not existing except for the precursor GGPP. And all aromatic crops, you must be knowing, GGPP is there. So that means any aromatic crops can be made into pharmaceutical crops. And food for thought. This is Mediterranean diet. People look at it and they feel, oh, so healthy. Nowadays, the generation which has come, millennial generation, they love this type of food. And just think of it, what does it have? It's basically a pizza. The base of pizza is not bread, it is eggplant, bangan. Bangan ka ek aapne chapati bana di. And then you are putting on that, broccoli and your cauliflower and your capsicum and many other things, which have molecules which are nutraceutically important. So that means for you all, those who want to work in this area, each component of such diets, which also happens in India, is possible to be developed into an industry. And that's what I would like to say, that a Nutri-Kisan can do it. A Kisan will say, I am a nutraceutical grower. I am not just wheat grower. He will say, I grow a very different kind of wheat which brings disadvantage to you. If you have gluten problem, eat my wheat. So that much education we have to give to the farmers. And in addition, biohorticulture gives you a very good hope. Biohorticulture, while we are talking of horticulture, fruits and vegetables, we usually talk of juices and pulps. But I say chemistry of that, again the whisper in fruits and vegetables. And if you do that, you will find whatever mostly you are throwing away after taking out juice or using the pulp or eating, what you are throwing, even like a banana peel and so on, that is what contains the nutraceuticals. So can we really do the processing? That's what I have discussed in this paper. It still holds very true, although it is now 10 years old paper. So therefore, if we want to do that economics and ecology, we have to think of resources and market. So we have to be global, not local or global. We should not, differentiate. Whatever is good in another country, we can take it and make it ours, and whatever is good in ours, we can give to them. Let them do it. Chakwe India, we have to pass on. And unless and until we do that, we are not going to make any sense. Today in the morning, Dr. Pentel was discussing with me why things are not happening. The things are not happening because we don't believe in passing on. We want that A to Z, I do. First thing. Second, we do not know end to end. Where do we start and where do we want to reach? We know only one or two terminologies which fascinate us, and we start calling ourselves experts of that. That's what has to go. So I just give you some very simple examples. So phorafane in broccoli. Why broccoli is healthy? Because of this. And that is a stimulant for enzymes which detoxify chemical carcinogens. So this is a nutraceutical. Now, how do you evaluate it and how you make product out of it is important. So calcium fortified food. Now, those people who have milk energy can take soya milk or many other milks coming from pulses, many kinds of pulses and even cereals. And those can really give you calcium as well. So all beans are rich in calcium. Vitamin and minerals. As I told you, what you are throwing out from fruits is vitamins and minerals. So you take only sugar and some of the energy from there or some flavonoids, but you throw the real thing. We have to take that. We can make that into animal feed, we can make them into human feed, infant feed, and whatnot. And then rest of the material also. It's a wrong trend of thinking that has developed that whatever is now getting wasted, we should make compost. You know, they say we have got a very good biocomposting or decomposer. Nonsense. You have to first take out all the things. What still cannot be handled, the fiber and other things, let it go into composting. Rest of the things will give you these industries. And if we start doing that, we are sitting on a wonderful situation. I told you about golden rice, beta carotene. Now here is an answer. If you cannot or you do not want GM, then also you have answer by combining horticultural rice. That means a few leaves of Moringa oleifera on steamed rice give you better than golden rice in terms of beta carotene content. Carrot gave you the name beta carotene, which is a precursor of vitamin A, which is good for your eyes. And carrot, 1 kilogram is equivalent to 32 grams of leaves of moringa and more stable form of your beta carotene. So that means that if we just start exploring this and my farmer says that I am a producer of vitamin A, what is the harm? The only thing is we have to teach them, make them learn it. And I found through my movement in villages since last more than a decade that the sons and daughters of farmers are able to understand very quickly. And they can be doing their entrepreneurship in a small room rather than only the field. So field, let it be with the farmer, his sons and daughters start doing processing, and that is what secondary agriculture or value addition is. Not only that, moringa not only produces beta carotene, it's a wonder tree. And one of the things that we found at CMAC was a bioenhancer. That means it doesn't have the drug activity of its own, but when combined with a drug like rifampicin, it enhanced its availability. And also we found that vitamin B12 was enhanced in terms of absorption in the gut, and that answers why in sambar we have drumstick. So that is the type of knowledge we have to have. And niaziridine was a molecule which my PhD student, Mr. Jayashankar Arya, could isolate up to and work on its mode of action, which is a bioenhancer in moringa. And next aspect is, let's not say we want to be only futuristic or we want to be modern. Ancient things have a lot of knowledge. And that ancient things, how we convert into modern platter is what we have to do, and that's the market of superfoods. Ancient wisdom of food says that the first crops which were called real good ones or most ancient foods that we have been eating is pulses or legumes. And you can see that legumes, they fix nitrogen with rhizobium in the soil. They are good for soil, they are good for plant, they are good for environment, and therefore eco-agriculture. And health and nutrition, protein, and the amino acid requirement that you have with bioavailability in a very good manner. You have examples if you want to shift from a non-vegetarian source to vegetarian. Soybean gave you one example, but now so many crops coming up. There is a report which came up in 2017, which talked about the emergence of these ancient grains as future. So ancient becoming future. And that's where pulses, as I already told you, there came another crop, which is pseudo cereal, quinoa, which is a cousin of Bathua family, was considered as International Year in 2013. And today we are talking of millets becoming 2023 as International Year of Millets. So that's going back into ancient, which we had replaced because of the abundance of wheat, rice, and maize system. So we could think in terms of that and start having the crops like millets, pseudo cereals, and many others. We have to understand another thing. There is a gut-brain connection. You know, we talk of central nervous system, we talk of peripheral nervous system, but here I am talking of the gut governed nervous system. That's where probiotics make a lot of meaning. Our gut contains almost 167 species of microbes, which maintain our health. And if we get rid of them or we disturb them, then our health deteriorates. And therefore, whatever you do with your genome, unless and until genome of these microbes is working, you are not going to get that efficient or proficient. And these proficiency comes from these, which are live microorganisms, which do that. One of those bifidobacterium, which is anaerobic, and lactobacillus, which is aerobic, are the good examples where products like probiotics are coming. But probiotic won't work without a prebiotic, because when you ingest a probiotic, it goes into your gut, but then is excreted out also, because your defense system and your hunt for nutrition from your gut doesn't let it get nutrition. And therefore, you need something which is not digested by you, or you are not using as carbon or energy source, but it doesn't cause you indigestion, and that is soluble fibers. So prebiotics again can be, as I was saying, a farmer can say, I grow prebiotics in my field. And that's what will help in developing symbiotics, and you get, healthy. And there are so many sources of prebiotics, right from fenugreek, chia, soybean, onion, garlic, oats. Oats, nowadays you take in the morning with cereal breakfast. That gives you mucilaginous thing, whether you put in milk or water. That's what is the soluble fiber. And chia is again a crop where, one hand it gives you the prebiotic, like here I am showing you the pudding. Now you just keep it in for half an hour in milk or water and milk together, and will add some honey or sugar. It becomes a pudding in half an hour, because it oozes out the lectomanins, which are going to be the prebiotics there. On the other hand, it is also a rich source of omega-3 fatty acid. And that's what chia is being grown for, as a superfood crop. And we also introduced in India, and the farmers are not only doubling their income, they have many times the incomes that they have got, in addition to their normal traditional crops. So omega-3 fatty acid itself can become a farmer's kitty, and there are a lot of options. What higher plants produce is ALA, but what your body needs is DHA or EPA. But you can metabolize ALA into DHA, and that's good for your brain, and also helping your cardiovascular system. And therefore, fish oil has that. But fish plant, you have ALA, and then you could make functional foods out of it. And plants which have omega-3 fatty acid is a huge list again, which we did not actually concentrate. We talked about flax and chia only, but then there are so many. Like you have linseed, what I talked about, corn, soybean, walnut, butternut, sesame, and rapeseed, which Dr. Pentel was talking, poppy, black bean, etc. And a movie, Jab We Met, becomes Jab We Millet. Now, when you do millet, then you can be meeting much better than Jab We Only Met. Okay, so we have been ignoring it. There is a caste system in crops. We consider this as inferior. This is a class which is not good for good people or wealthy people. No, now wealthy people want it. Again, the total thing is turning. And Karnataka state has taken a big lead. And we have a millet network of India centered there, MINI, in 2007. And the good thing is that if we look at the world, we are the predominant users of millet still in our traditional or folklore populations, and also producers of that. So why can't we make it into a mega thing that will happen in 2023, which I am telling you, International Year of Millets is going to perhaps open up the doors for that. And these are the 8 minor millets which you have. So if you see on right side here, little millet, tall millet, so you see the advantages: digestion friendly, high calcium, high protein for pearl millet, finger millet, ragi, which is little dark in color, is good for diabetic peoples and anemics, even iron source. Foxtail millet, which is good for immunity. Barnyard millet, which is rich in antioxidant and iron. Kodo millet or verago, which is high in fiber and very good again for diabetics and weight loss. And sorghum, which usually jowar we use, which is making very good chapati. So, making combination and making it a good formulation of a food, functional food, is feasible with millets. And that is how we will not lose on taste also. See, if I make a very good thing for you, which is very good, very healthy, and it doesn't smell good, or it doesn't taste good, or it doesn't feel good, then you are not going to eat, particularly children. And therefore, you have to do research in this area, and that's called functional food formulation. Another group of plants which farmers can grow are pseudo cereals. Now, cereals and pseudo cereals, the difference is that in cereals, the embryo is located in starchy endosperm, and in pseudo cereals, you have a perisperm which surrounds the embryo. And you have three very good examples. Quinoa I had talked about, buckwheat, that is kutu, the atta that we eat in Vrat, and amaranth or ramdana or rajgira. Now, this is a table which I am not going to read, but you can see that the possibilities of functional food industry can bank on it. You talk of any product that children like, you can make out of it. Whether it is kurkure type of thing, it is chips, or it is wavy materials, extruded products, and so on. And also sattu and other type of things are possible. So not to forget, when we are talking of ancient, that wheat and rice also have ancient history. They also have nutritional scope. So ancient heritage doesn't lie only with what I talked so far. You might be thinking that I am saying don't eat wheat, maize, rice, and eat only these things. No, go back to their history. And the history is the wheat which we know, the hexaploid, originated from two diploids crossing and then tetraploid crossing with another diploid, and then you get hexaploid. And those were earlier names spelled Faro, Kamut, and Farik. And these are the ones. Spelt is like this, Binkel wheat also we call it. It's rich in vitamin A, very good for heart, niacin rich, and it has very less gluten content. And this is farro, which is spelt wheat. Again known for its, you want to eat as a snack, this is very good. It has a nutty flavor. And then also has vitamin B3 and zinc. And this is kamut or Khorasan wheat, which is also called camel tooth or prophet's wheat, because of various usages traditionally that we had. And this is Triticum turgidum, or durum wheat, which makes very good noodles and pasta and other things. This comes out from this tetraploid wheat. And that's what I'm saying is that don't even focus only on one type of wheat or one type of rice. You have plenty there, the diversity there. Kala namak rice in rice. Now I was happy to see Dr. Ashok Kumar Singh, director of IARI, on Facebook telling about Kala Namak research that they are doing, and he's talking in farmers' fields, a good line doing there. It has been pending for long. Now, Kala Namak is a GI for India. It is also called Buddha's gift to Siddharth. And that's what has aroma as well as a black beauty spot on its grain. And it gives very good taste, a slight pinch of saltiness. So your biryani or your pulao is ready straight away. And that is what Kala Namak can do, and it can beat basmati. And that is how it looks, the Kala Namak rice. And then there is a black rice also. It was called forbidden rice in China because it was meant only for rich or royal families. But today it is available in Manipur. Manipur ki kheer, if you know, that is from black rice only. It's so rich in flavonoids, iron, and fiber, and many vitamins. Bhutanese red rice. That's again a very good thing, and you get in Kerala and even in Kashmir the land races which resemble that, and the advantages that you can have. And this is gluten free, so therefore those who are celiacs can have this very easily. And ultimately I come to, because I come from CMAP, in my history I will come to aroma crops. Aromaceutical farmers also are possible. Again, aroma goes into many therapies, including aromatherapy, where you could have a cultivation of health. And this is Kosi variety of mentha, which was grown by farmers in 80% of the cultivated in India, and it led to entrepreneurship in them, and they had distillation right in their fields and making markets straight away. So they will keep the canny of oil in their kitchen, and whenever they need, they will go and sell it. That means it's a currency in itself, the mentha oil, and the whole family gets involved. So this lady is harvesting from the distillation unit the mentha oil, and people move from kacha house to pakka house. So transformation it brings, economics it brings. And when also pharma crops are possible, if aroma crops are possible, and example I want to give, because that is where Dr. Abdin also has interest and has been interacting with CMAP on artemisia, which produces artemisinin, and artemisinin converted into arti-ether, artemether, artesunate, artilunate, many derivatives which can give very good form of the drugs against malaria when you have falciparum infection or multi-drug resistant malaria. We use here typical molecular breeding approach of mass recurrent selection with markers that get related with high yields, which is related with the biosynthetic pathway of synthesis of artemisinin in its metabolism, and came out with a variety called Sim-Marogya. Sim is from CMAP, which really produced from 0.2% of artemisinin, we reached 1.2% or 1.1% of artemisinin yield, which changed the whole scenario. Earlier, our cost of production was 40,000 rupees per kilogram of artemisinin. China was selling in the world 16,000 rupees a kilogram. With this, we brought it to 12,000 rupees a kilogram, and we export to 42 countries and could translate this technology to many industries with full package of agriculture and processing. And these industries, first one which you see on the left side top, Thimis Healthcare, Dr. Dinesh Patel, who was also a product of CDRI as a scientist, but he is owner of this company, has been giving crores of rupees as royalty to CSIR because of this technology. And 42 countries they are exporting, Sub-Saharan Africa particularly. And the whole technology package ultimately led to chemist shop with the name EMAL, that is eradicate malaria. Email came later, EMAL came first. And this EMAL is eradicate malaria, which is arti-ether, alpha beta arti-ether mixture, which is in oil base, and we have to give intramuscular injection when you have a patient. And then question was, can we make it water soluble? Because it was not water soluble, so it has to go into oil base. We created artesunate, artilinate, and many other derivatives. And ultimately, I come to my passion, which I'm following now, is bio-villages. We created at CMAP, we can create e-villages. E-villages means enabled, entrepreneurial, economical, economy empowered, and eco-efficient. And that's what we created Flora Fauna Science Foundation in 2015. Now it's 6 years of that. And we have enabling society to achieve health and nutrition as its tagline. And the vision is to create so many e-villages which farmers can really take up new crops as entrepreneurs of specialist kinds. And this was first Rajagada in Itonja region, where we not only gave them some technologies, but also machinery to do the harvesting without use of any biofuels or any petroleum thing. No tractor needed. One person can do equivalent to 8 people with this sickle. And this sickle, we had a farmer from Canada coming here, and we demonstrated that. You will not imagine, in the demonstration itself, half of the village wheat had been harvested in half day. So that's what can happen. This was Chia village we created in Ahmedpur in Raebareli district. And then ultimately I come to how I can help you all, the students and even the scientists who want to be entrepreneurs. So the thing is, let's bring that change coming from ancient to future. The products are possible. So if the farmer is producing, farmer's son will be doing, you can do high-end technology part, and that is possible. If you would like to join in my incubator, I call it not incubator, incubator. And incubator is I incubate the innovations. So innovation incubator is where you could come. There is no charges for that. You can come and work and establish yourself as entrepreneur. And this is food innovations is a venture where people have already started making products, and with the cooperation of farmers and industry, I could raise even 7 to 8 startups who are industry today. And the products like this is Yo5 chocolate bar, which is rich in yogurt protein and fiber, and therefore healthy chocolate. You have vegetarian protein bars for gym goers and bodybuilders. Here you see one of the actors who is known for it. And when he took in a show where we were launching our product, he liked it so much that when he was performing, he took our bar and started eating there. So we took his picture. So this function was body power. And the Nutra High products, we created functional noodles, pasta, and cookies, which have several of these things which I told you. And this is DG of CSIR, Sameer Brahmachari at that time, and director of CFTRI, who were there at launch of this. This was the latest I did, NutriPlanet, a company that I raised from scratch, and they are doing good in chia and nutrition value chain. So they could create these 2 bars, and ultimately now they are even exporting things. So what do we expect? Consumers are demanding more from their food and are looking for more ways to get nutrients and compounds. They do not like pills and capsules. So if we get this psyche of the world and bring it to the farmer, farmer can make things for you. And therefore, future agriculture or future of agriculture we have to talk of, I would say it is eco-agriculture. You know, so many terms are being given, organic agriculture or natural farming, and so many things. I don't believe in that. I believe that it has to be economically and ecologically balanced agriculture that has to come up, and that we have to understand why it is not happening. It is because we are skewing ourselves. We are not maintaining the biodiversity by, you cannot do biodiversity maintenance by telling people that you create a gene bank or you create a biodiversity park. It has to be economically rewarding. And if you want to do that, as I told you about ancient grains and superfoods that can come from ancient knowledge of folklore usage, you can create eco-agriculture. And this will be regenerative model. If you keep more diversity in your fields, more and more species, and within those species, more and more diverse kinds, then you are going to keep the pests and insects or diseases at bay. So you don't even need chemicals to be used. But don't say that, there should not be any use of chemicals. Even plants produce chemicals, as I told you. So chemicals, we should not be having reservation. Chemicals are way of life, but which chemicals, what chemicals, how produced? And there is an enormous diversity loss. I give you this WWF report. 75% of food humans consume comes from only 12 plant sources and 5 animal sources, making up to 60% of the plant-based calories in entire human diet globally. So just imagine, so many species, we are doing only 12 plant sources mainly. So this also tells that, as an example, 75% of genetic plant diversity has been lost in Thailand. They were having 16,000 rice varieties, and today they have only 37. So the thing is, the challenge for agricultural scientists is bring those land races, raise their yields and adaptability they already have. So you don't need GM in that case. You can straight away have the answers here. And repeated harvesting of the same crop on same land makes it empty. We should have so much of diversity available with us. And as I told you about the secondary agriculture, the products made out of that farmers will be happy to do that. So I'd like to acknowledge the sources of information which I have used to reach to you on these aspects. And also a statement by Darwin. It is not the strongest of the species that survive, not the most intelligent, but ones most responsive to change. I was hearing in the morning a statement that a person working on wheat should spend the whole life in wheat, and a person working on brassica should spend whole life on brassica. No, I don't agree with that. Person has to change. Person has to come according to situation that is demanded. That's why you are scientist. You are not a scientist of only one field. Scientist means what and why answer. So give what and why answer to whatever situation demands from you, and you can create wonders. I acknowledge this team of CMAP, which walked with me on green paths to better health and life, around 600 people, 100 scientists and supporting staff and students. And these are the industries that I could raise through translational R&D. And this is my introduction. My introduction was given earlier, but I would like to believe this is my introduction. And I love it. And my dream is Prasanna Bharat. And the route that I have followed is Khet se Sehat, Poshan se Roshan, and today Krishi se Khushi is going on. So thank you all.
Wow, such a thought-provoking lecture it was. Thank you, sir. This talk will definitely help us to think out of the box. Now is the time. Is there any question from the audience? Any discussion?
Can I give you 2 minutes? Summarize it? It's wonderful. You must be agreeing with me that this. I'll take at least 2. Okay. Summary. So don't worry. 2 hours. It's okay. You must be agreeing with me that this is a wonderful treat of science today in the morning. And the 3 means legends. Means you can say that they are the known people in their own field, and probably when they are in full flow, it is very difficult to stop them. And when they are your teachers, when they are your mentors. So in the beginning, we learn about the Barnage and Barster, and he is the first person for this particular transgenic system was being approved by GEAC. We know that this is from India, from the crop plant. Dr. Pental, wonderful lecture. Then after that, we listen to Dr. Sudhanshu, and he is the person again involved in the rotavirus development, the vaccine development. Again, he was in the full flow. I'm sorry, sir, in between. But sometimes it is very difficult. He has put me as a chairman, and when I am sitting as the chairman in front of Dr. Khanuja, it is very difficult to I cannot sit there because he is my teacher. He has taught me molecular biology in IRL, and whatever I am, and I was working with him in CMAP under his leadership. Whatever I am as a scientist, I have learned from him. The basic molecular biology is clear from his teachings only. So I will not give much more introduction about him. And what he has told about, he has given a full package, starting from the science, different plants, nutraceuticals, and whatnot, and how you can go for entrepreneurship. And till now, we are working together in some of the aspects, and we need his blessings. And we'll be definitely, sir, you will be giving your blessings to us in future also. Go on giving. I'm with you. And as I told, this is the whole summary, and now I can take over. Thank you. Thank you.
Thank you, sir. Now is the time to felicitate our distinguished speakers. Professor Dept.of sir had to leave because of some important engagement. So I request the second speaker to please come up on the stage. I request the chairpersons to please felicitate Professor Sudhanshu Vrati. Now I request the chairpersons to please felicitate Professor S.K. Khanuja sir. Now time to felicitate our chair and co-chair. I request Dr. Rima Pant ma'am to please felicitate the chairperson of the session, Dr. Ajit Kumar Shastri sir. And now I request Dr. Ajit Kumar Shastri sir to please felicitate Dr. Eva Bandhan. Thank you so much. So this was the end of plenary session 1. This is the meal time now. So we will be resuming after the lunch. So enjoy your meal. Thank you.
On live. Okay. Actually, we are starting our session after lunch. So you must be feeling sleepy. So if you sleep opening your eyes, so that I will understand that you are listening to me and I will be encouraged, more and more encouraged. And last time I spoke here, I told I think in India and convert to English, Hindi, then translate to English. And now I have learned little bit, so I'm in between portion I have taken out. But I think in India and directly translate to English and speak to you. So quickly, because our time is very limited, so I'll speak about genetic enhancement and nutritional security. So we know that from the Green Revolution until now, we have tremendous increase in food grain production. So if you see the net soil area that has been increased not so that it can be counted statistically. It is not that that much increase is there, but productivity has been increased. If productivity is increased and net soil area remain the same, so the you say that there is something happening in between from the Green Revolution till now. So there is continuous improvement in the genotypes. So we have milk production, fish production, crop production, horticultural production, those all are increasing. Productivity is increasing. So whether we need food security or nutritional security, that's the question. So 8.9% of the world population still lives in hunger, we know that. So we need food security, that's for sure. We always want food. Our population is increasing and we need food. So to meet this food security, we have to increase the food grain production 70%. So these are the general data spoken by everyone. Climate change, again people they speak about climate change, that's a reality. Well, it is affecting the crop yield, that's a reality. I'm not going to detail because 20 minutes, because otherwise Dr. Panda will be coming in between. But there is diminishing availability of protein, iron, zinc because of the climate change. Because plants they are absorbing less, it is coming to us less. Article 47 of the Indian Constitution speaks about food security as a nutritional security, that we have to provide food, better food, nutritious food to our population. That's Article 47 directive principle. So ensuring food and nutritional security, we have to ensure both the things, food security as well as nutritional security. So for that reason, we have to increase the yield of the plant. Also, we have to induce nutrition inside the plant. But is it true? We are doing it. Try nutrition then. So these are the daily nutrition. It includes vitamin, amino acids, different metals. So all these things we need in our body. So these are the nutrients. But from Green Revolution till now, what we are doing? We are only focusing on yield and yield. So in this, a breeder, I have learned from my teacher, Dr. Khanuja, a breeder is a breeder who rejects maximum of the things. Only focus on one thing, the character, which he wants. So we wanted food because people were starving at that period of time. So increased our food production, we only concentrated on yield. And we forgot about the nutrition. If you go to your home, so if you in a village, so you get some indigenous variety to eat, but the tastes are different. The taste different. So those tastes you are not able to get right now. You get very polished white rice, polished food. Always you like also polished food. So this is happening. So there is a nutrition, and Vishwanathan will be maybe talking later on about this, how to go for genome editing in detail. And this is an example. So the production has gone up in rice, but G concentration in rice variety has gone down. And this is the data which has been published from ICR data by our current DG. Due to climate change, how it is, what is happening, how nutrition is going down. This is iron content of rice. Again, it is going down in different variety. We are selecting variety for high yield, but the content of different metals are going down, which you need.
So which are the desired traits in crops? We want high content of quality of protein, high content and quality of oil, high vitamin content, low toxic substance with harmful human health, and biofortification. These are the buzzwords right now we are listening. And we know there are 20 amino acids and there are 8 to 9 amino acids, these are essential. And we know the biochemical pathway. We know the genes. So why can't we right now go for intervening in those particular genes to increase the essential amino acids in the crop plant? Can we do that? Yes, we can do that. We are doing it. So these are the amino acids. The status of elements, these are different elements again we need. There are different transporters. The problem in the crop plant is one particular metal uptaken by the plant, the transporters, it may uptake some heavy metal also. It may uptake some deleterious metal also. So how we are going to separate the uptake of metals? We want good metals to be uptaken by the plant so that you can eat, and it should go and directly deposited in the part which you eat. If it is deposited in some other part, then this is no use to us. So these are the type of research, and we are working in this direction. Biotechnologist. These are the metals I told. Again, there are different vitamins. Again, the biochemical pathway of vitamins we know, the genes we know. So can we enhance? It's not a $1 million question that we'll get the gene and go for the target. Targets, genes already there, are being there. So some of the students, some of the people can go in this direction in different crop plants to increase vitamins, amino acids, and all these things. So these are the pathways. I'm not going to detail. Quickly, I'll go biofortification of iron and zinc in crops. This is the ICR data. So there are several crops that have been developed in ICR by biofortifying the crops with iron, zinc, and in wheat, pearl millet, lentil, pomegranate. So the variety has been developed. Around 87 varieties are being developed till now. Again, 18 varieties or 17 varieties are added last year. So these are the biofortified variety are coming up from ICR. So these are again the data in different horticultural crops and different food crops. So transgenic and genome editing. So whole lot of characters we can intervene. So I have listed those characters. And if you see the genetic enhancement, if you go for see the ancestral crop, so we got the recent crop through spontaneous mutation, which is again more than 1,000 years ago. Then traditional crop modification happened 10,000 years ago. Then random mutation. Then precise mutation. Precise mutation is genome editing nowadays we call. So we have got the SOPs, we have got the guidelines ready, and go for it. And there are different, we are getting our thesis from different part of India, different universities as well as from ICR. So people are working on genome editing. Many crop plants are being intervened like tomato, rice. Dr. Vishwanathan, he is already successful in rice. He will be telling you about that. So there are many, in our institute also people are working on mustard and pigeon pea. So many crops will come in near future, intervening in this different genes to get better product with highly nutritious product at a high yield. Again, we are talking about the oil seeds. We are importing lots of oil into India. So this genome editing will definitely help us to make Atmanirbhar Bharat in future, which we think, and we are working in this direction to increase the grain weight of the plant, to increase the silica number of the plant, to increase the number of branches so that they can bear silica, to increase the strength of the plant so that they can bear more silica. So these are the characters we are working on. And again, you cannot ignore the quality. What type of oil? How much unsaturated oil you want? How much saturated you want? And industrial demands and our demands, eating demands are different. We want more omega fatty acids, but industrial demands they want the oil which can withstand the rigor of high humidity. Industrial processing, rigor of industrial processing, which includes high humidity, high temperature, high pressure. So these are the balance we have to make, which type of, which direction we will go to breed the crop or to intervene in the crop to get good character in the crop. So this is the list of, from the ISA side, this is the list of plants, around 32 plants, those are being developed worldwide for different characters, transgenic plants. And mainly these transgenic plants are Bt toxin, maybe herbicide tolerance, or maybe somewhere in like some sugar metabolism in maize and some other carbohydrate plant. So the characters are limited in transgenic plants, in crops are limited. In 32 crops, people are working. They include horticultural plants, they include fruit plants, they include medicinal plants. So some of the things are being done. And in India, we have cotton. Cotton has been approved by GESC earlier. That is again soybean. It has been for their import. We have to import because we are good quality soybean not available in India for animal feed. So we are importing it. And recently, Barnage Baster, that is by the Dept.of Pental, which has been approved by GESC. Again, Madam Ahuja, she is also instrumental in working day and night on that for the approval of this particular plant. And this is till July 2022. Again, these are the plants which are for different characters. And you know about the genome editing Nobel Prize and the timeline. The timelines: herbicide resistance crop, blight resistance rice, then herbicide resistant canola, powdery mildew resistant wheat, then high yield waxy corn, soybean with reduced trans fat. There are many things. After domesticated tomato, after intervening into 4 genes or so, the wild type tomato becomes domesticated. Good quality tomato, cassava, cyanide free cassava, low gluten transgenic wheat. These are already being developed. And one or two they have come to the market, like Gava tomato from Japan. Simply intervening in single gene. Also, there is an important thing in rice, the uptake, iron uptake. So intervening in one gene, the mechanism is not known, but instead of depositing iron in the leaf seed, deposit it in the grain. So we have to have more mechanistic insight in the science. What is happening inside the plant? It is happening, but still then we need to investigate more in this direction. So we can get vitamin D3 by intervening in something called cholecalciferol biosynthesis. Cholecalciferol is the D3. Cholecalciferol, when you sit in the sun, then it is converted to vitamin D3. This is produced from the triterpene sterols. So those are the intervention can be done, and some of the target being used in CRISPR-Cas. Your right side column, you can see that in India we are using target. There is no way of India. Either you are following the literature or following the patents. Maybe 50% of the targets are there, IPR protected. Again, genome editing is IPR protected. The technology is IPR protected. So we have to spend much more as a country if you come out with a product in future. So better thing will be that we have to find out our alternative targets. I told the pathway is known. It is in the public domain. This is not IPR protected. So many things are being known, and we have to do research in this direction to get good targets in the genes. We have the genomes, sequence the genomes, and from the genomes mechanistically we will analyze and find out the targets. So can we, as a country together, can we have targets, a repository of targets? IPR protected. That will be utilizable in future. We are working in this direction in an IPB again. So the focus are many things, bacterial, viral, fungal diseases, like I said, and there are many things, the economic characters you can, people are working again, big Melinda Foundation, converting the plant to super plants. So can we afford ignore the technology advances in agriculture? Previously we were pulling the cart by horses or bullocks. Now we have tractors. So can we accept the transgenic plants in future? Can we accept the genome edited plant in a better way in future? We have to accept. And we have to accept, why I told you we have to accept, when people will start starving, definitely we will eat. Just like your corona vaccination. The vaccine has come quickly. Rotavirus has taken a long journey. Coronavirus, without testing much, we have injected inside us because we were dying at that time. So when we will start starving, we will eat the transgenic plant, rather die. So this will happen in the future. So all the organizations all over the world, they support transgenic plants. So genetic advanced yield and nutrition, people are doing it. There are different type of animals, genetic interventions are going on. QTLs, we know about the QTLs. Can we analyze the QTLs and find out mechanistically which are the genes involved in the QTLs? Precisely. Those things we can do, we are doing it. Again, genome editing will rise. I have already talked. Plant immunity. Again, I will take a few from you, sir. This Ali Sart tells that secondary metabolites in armory. So they are standing, I have learned from him. He speaks always. The plants are outside, they are facing sun, they are facing everything, they are facing different type of stresses. But how they are protecting themselves. And this, he has shown the picture, and I have a little bit converted it. This picture. So in the plant's armory, these are there to protect themselves. And we always start, I have marked one thing. We will say, we will introduce the plant with different type of QTLs, a long chunk of DNA we introduce. But when you analyze the DNA, one or two secondary metabolite influencing transcription vector or pathway genes are there. Just imagine, why paste of mustard cannot attack rice? Why a paste of rice cannot attack wheat? But it may be possible in due course of time when the climate is changing. Climate change is not the effect of simply stress, a single stress, water stress or high water stress or a heat stress. It is not that. All the stresses will come at unknown period of time and stay long for duration. That is climate change. It is going to happen. And we should not do research only on single stress. Together, we have to approach them together. So these are the metabolites they will help in future. TDA आए थे, सिनाप गए थे, सिनाप में खा लिए थे सब खा लिए थे, लेकिन मेडिसिन के बारे में प्लांट बचे हुए थे. तो उनका अंदर कुछ है. These are the tulsi plants. This has been developed in. I will not talk about much about that. We have sequenced the genome of tulsi. There are many molecules in that. We have different genetic strategic genome resources. From there, we can isolate some of the genes. These are the genes already being isolated by our lab. Monoterpene biosynthesis gene, sesquiterpene biosynthesis gene. There are, I'll go quickly. Delidipural, you see the amount. 10 milligram costs around $300. It can express from 0.5% to 0.5% to around 25%. So the fractionation will be easy. Synteny, another concept. That is synteny. The pathways are existing in all the plants. The secondary metabolite pathway. But it is dominant in one plant. And, dominant means I am not talking about the genetic dominance, the production dominance. It produces highly, the amount is much more in. I'm going quicker for that because of Dr. Panda. Otherwise, he will not teach me. So there are different molecules, they are present in different plants. So can you reduce these molecules? So one example, cis-3-hexanol, some small molecules, they regulate the immunity of the plant, not only our immunity. We learn from the plant because plant is protecting themselves while standing and facing different abiotic and abiotic stresses, and we are using those molecular our medicine. Why not plant will be using their own medicines also? We have to think in that direction. So this is cis-3-hexanol. This induces teloge, and teloge actually prohibits the viral replication. So this happens. There are literatures, and this is our own example. Then Urania sominifera, if you one gene, that will be inhibiting the Pseudomonas in potato. If you reduce the farnesyl biosynthesis, then it will be inhibiting the lead blight, the fungus. So there are different, and again, this is again from our own publication that Heliothis arnigera, this can be inhibited by some of the pathogens, specific pathogens. So we can get the knowledge from the wild type relatives. Why they are not infecting the wild type? Definitely, the plant which is not host to a particular insect and pest or the fungi may be the host in future. So we have to think of the future. In which direction? We have to find out the trend. So this type of what we have to do. So this is a major achievement. Again, Pusa Jaikisan and all these things which are being developed at NIPB. Around 12 genomes we have sequenced already in NIPB. There are different varieties with collaboration with others we have done. And now GEC has given approval to go for the field testing of one of the fusion BT genotype. So it will be tested in recent future, but this time it cannot be tested because the season is up. So we are asking permission for the next season. So what is the future way forward? So we have to have a build up a repository of targets which are IPR protected, genome editing, transgenics. These are for the future, and if not, then you have to find out the internal immunity of the plant to induce them to face the climate change in future. Not only food security, for nutritional security also. Thank you. Thank you, sir. I think I have completed within 20 minutes. 20.
Thank you so much, sir, for this valuable talk. This work will surely help us to provide a better nutrition and to withstand the environmental challenges that might otherwise wipe out a whole season of produce. Now we are having a great opportunity to listen to the words by a great knowledgeable speaker, Dr. Vishwanathan Chinnaswamy. Sir is a joint director at ICR Indian Agricultural Research Institute. Dr. Vishwanathan has received his postdoctoral training from University of Arizona, California, and Howard. He established state-of-the-art automated plant genomics facility at IARI and has various research publications and awards to his credit, such as J.J. Shenoy Gold Medal from Indian Society for Plant Physiology and many more. He is a fellow of National Academy of Agricultural Sciences, fellow of Indian Society for Plant Physiology, and associate of National Academy of Agricultural Sciences. It is my privilege to invite you here on stage, sir, for your visionary words in the field of genome editing for sustainable food security.
Ajit, sir was my teacher from whom we hours. And 1 hour before we have to go and write whatever he has written in this big book. So such a wonderful teacher, sir. It is a privilege to present before you. And I thank Professor Abdin for inviting me for this lecture. Dr. Sasani, Dr. Ogeba, Dr. Reddy, and all the distinguished speakers and participants, my dear students. So I am from the Indian Agriculture Research Institute, and many of you might be knowing this is 117-year-old institution. It was originally started in Pusa, Bihar in 1905, and in 1936 it was shifted to New Delhi, and it is known as the seat of Green Revolution in India. And we are also offering higher education, and from this year we are starting undergraduate education also. From 1923, the institute is offering higher education, so almost we are going to complete 100 years. So today I will be talking about genome editing for sustainable food security. Mainly I will be focusing on rice, and I thank Dr. Sasani for making my task easier, so I need not give the introduction. We know why food security is important. And just I want to say, because I will be making my presentation mostly on rice, so I want to say why rice is important, because it is contributing to about 40% of the food production in the country. Besides, it has a lot of export value. And in the future climate, we have to keep our environment sustainable. So under that context also, we have to reduce methane emission from paddy. So in this context also, rice is very important, and it is one of the crop which uses more than 50% of the water. So if you want to save water in agriculture, we have to save water in rice. Either we have to make it direct shaded and drought tolerant. And this already, we know that a lot of civilizations have disappeared because we did not care for the environment and the stress resilience of the crop. So I'll be talking about genome editing. Already my previous speaker told it is precision mutagenesis tool. So mutations are the basis for evolution and crop improvement, which we all know. Just I want you to recollect these two mutants. One is the mutation of wheat, which is the reduced height one, a dominant mutation that led to gain of function of the GALA protein that reduced the plant height. And another one mutation is this is the mutation in GA20 oxidase gene, which made the loss of function, so the plant produces less DNA and it becomes dwarf. So these two genes are feeding the world today. If I say it is not an exaggeration, because of these two genes, we have got great revolution in the rice and wheat, and because these are because of the natural mutation. But these kind of mutations occur once in a while. So later the induced mutations are coming, and because scientists were looking for the way to create the precise mutagenesis, and most of you might have studied, all the biology students might have studied about the Muller and Stadler and their experiment. If you see all these mutations, mutagenic agents, they cause DNA damage at random in the genome. So once this DNA damage gets repaired, you get mutation. So you remember that if we have some tools and technique to precisely create a mutation or precisely damage the DNA, then it is possible to induce mutation at precise sites. So these are the earlier development. Some of my colleagues who have studied at the earlier time might have remembered the gene targeting technology, which was very cumbersome. Later, these three technology came in, that is meganuclease, zinc finger nuclease, and tail effectors. All these three are dependent on the manner in which the protein can recognize the DNA. Just like restriction enzyme, they can recognize the DNA at specific sequence, and if you attach a nuclease activity to it, whereas the meganuclease has its own nuclease activity, whereas the zinc fingers and tail effectors, they do not have nuclease activity. So we attach a nuclease to it so that we direct the protein to go to a specific location on the genome, and the nuclease cut the genome in that particular site. Then repair process leads to creation of mutation, and this involves protein engineering, which is relatively a difficult task, and it is difficult to engineer protein to target each and every sequence of interest in the genome. So hence the advancement came in, which is an RNA-guided genome editing. That is, it utilizes the RNA complementarity to the DNA. The complementary base pairing is utilized in this. So an RNA molecule can guide the nuclease protein to a specific location in the genome. And this invention made by Jennifer Doudna and Charpentier received Nobel Prize in 2020. Most of you might have studied about this. So the genome editing needs two reagents. One is the guide RNA, which takes the nucleus or the molecular scissors to a specific location on the genome, and the molecular scissors per stick. So these two tools need to be introduced into the plant cell either through genetic transformation or through protoplast method. And then once it reaches into the genome, the RNA is produced from the RNA polymerase III driven promoter, and Cas9 is produced from the Pol2 promoter. And then this protein and this guide RNA makes a complex. It goes and recognizes the target sequence. Wherever we have designed the guide RNA, which has complementarity to this region, it goes and binds to that place. And this Cas9 enzyme can cut both the strands of DNA, so it makes a perfect cut, and it is about 3 nucleotide upstream to the protospacer and tissue motif. So once these cuts are generated, it can be repaired in 3 different fashions. One is the template, without template, it is unguided repair. Once the DNA cut is made, we are not directing the cell, and the cell takes care of the repair, which uses non-homologous engineering pathway, which we call it as site-directed nuclease 1. The other one is we can provide a repair template. That is, we can guide the cell to take this DNA as a copy to repair the cut site. Wherein the repair template is similar to that of the native template, except for 1 or 2 desired nucleotide changes that we want to introduce. So this kind of mutation is called as SERB mutation. And the third one is the SDN3 mutations, where besides the homology arm, we use DNA which is foreign, which can go and integrate into the cut site. This is also very important in case of we want to develop transgenics. We can target the foreign gene into a safe harbor loci. So we need not produce 5,000 event. We can produce 5 or 10 event to get select a good event. So these two are considered as the product derived from the SDN1, and SDN2 will be considered as non-GM, and this one will be GM. So Dr. Vibha Madam will be talking you in detail about this, how it is regulated. So some example I would like to give before going to my own work. So you remember the domestication, which is a very long process, and this is one example where the shattering proved gene one, which if it is normal, it gives shattering. If we create a mutation, it gives a non-shattering phenotype. This would be created in rice. So the large years of domestication event can be generated within the generation by using genome editing. So similarly, the green revolution mutant which I talked, which can be generated easily by the genome editing. Even the wild type Borisa alta can be converted into normal rice by using a simple genome editing. And another example I want to give you is the herbicide tolerance in rice. Because of the DBT efforts, almost it took 10 years to identify one mutant which is tolerant to herbicide in rice. And this can be easily created by using the genome editing. So the amount of time and effort which is required can be reduced quickly. So this example is already Dr. Sashni told about this one, Vaxicon, where we produce a corn with almost 100% amylopectin, which has very high industrial use. And when we talk about genome editing, many students think that it can be useful only to create loss of function mutation or knockout mutation, or just we want to modify one or two amino acids. This is an example wherein we can alter the expression of the gene only under the specific condition. So this, in this case, you use promoter editing. This is an example of bacterial blight causing pathogen. When it infects rice, it produces an effector protein which go and induces sweet genes. So if we know where the effector protein goes and binds to the promoter, so only if we knock out this particular cis element, then under the pathogen infection condition it will not be induced, otherwise this gene will be expressed. So by using this, bacterial blight resistance has been engineered in rice. Similar, another one, promoter editing, is ideal plant architecture one. You can see the yield difference that you obtain by editing the promoter region by enhancing the expression of IPA1. This example was also touched upon, where you can convert Solanum pipinellifolium, which is a wild tomato, into a cultivated tomato within a short period of time. And you can see the fruit size quickly can be increased to a normal size while training all the resistance. And another one is also earlier touched upon by my previous speaker. So the vitamin D rich tomato. So we need not worry about if you are totally non-vegetarian, you have an alternate source of the vitamin D. If you see the global scenario, this is the 2020 report. Almost 231 product-oriented publications have been made, and these are the trade that has been listed. And if you see the crops, rice is the most worked out crop because of its importance in the food production and importance in the climate change, and also availability of the genome and known genes in the rice as compared to other crops, which is followed by tomato. And almost about 168 products have been given the non-regulatory status in USA alone. In ICR, all institutes, most of the institutes, they are working on genome editing. Once the favorable regulation came from the government, many young scientists, they got interested in this one. And these are the various crops that ICR institutes are working. And these are 26 institutes are working, almost about 75 scientists are involved. And these are various traits for which scientists are working on genome editing. And this also I told, like, we need not only create the knockout mutation. Depending upon the target site, we can create all kind of mutation. In our lab, we are working with different genes in the project funded by NASF and project funded by DBT. So today I'll be mainly talking briefly about the DST mutation. So this mutation originally in an EMS mutagenized population, it was discovered in the China in 2009. The mutation in this particular zinc finger transcription factor, which makes it loss of function, gives tolerance to drought and salt stress at seedling stage. We also looked for similar kind of mutation in the Nagina22 mutant population, but we were not successful in getting a mutation in the DST gene. So even if you know the mutation, it exists in some spaces. It is not easy to get by using the conventional mutagenesis. As I told, the mutation events are rare with reference to the specific gene. And another advantage of the CRISPR tool is that if you know a gene which is conserved across the species, if you know the function in one plant, you can edit in the other plant, and you can get the similar function, which is recently demonstrated in several crops. So we selected this DST gene, and we designed guide RNA for 2 different places. And the vector for this I have obtained from Professor JKC, who is my mentor when I did postdoc with him. Almost I was with him for 3 years. And Santosh Kumar is the one who carried out this particular work. So in T0, we obtained about 30 different events, and when we sequenced, we obtained about 19 different mutants, and these are the different alleles that we have selected. So this is the PAM site that I told, and about 3 nucleotide upstream, the nucleus cuts, so you expect mutation around this place. And so we have got either single base pair deletion, 2 base pair deletion, and then cut occurs at both the guide RNA site. You get a large deletion or the mutation in the second nucleotide. So all these create different loss of function mutation. So this is the change in the protein of the wild type protein versus the different mutant version is shown. So all create a loss of function mutant. So earlier it was shown that this protein reduces the water loss by causing the stomatal closure, and we examined these different alleles. We found that they also reduce the stomatal number, thereby reducing the water loss, and it is evident in the water loss assay also. So to understand the function, we examined the genes involved in the stomatal development, and we found that at least 3 of the genes involved in the stomatal development are less expressed in the mutant, and 2 of the genes, they have the zinc finger transcription factor DST binding domain, which means the mutation in this loss of function causes reduced expression. And as demonstrated earlier, these loss of function mutants also provide high level of tolerance to salt stress at the seedling stage. But our interest is on the reproductive stage stress tolerance. So we examined under 200 millimolar NaCl, both the wild type and mutant plant planted in the same pot. We found that the mutant plants are more tolerant, both under salinity as well as under the drought stress condition. So many earlier lot of worries were there about off target. So we analyzed all the predicted off target by sequencing, and we did not find any off target mutation in the selected mutant. And it is also important that whatever variety we develop, it not only should be climate resilient and yielding high under stress condition, it should also yield high under the normal condition. So this mutation, all the 4 different kind of mutations that we generate, it produces more productive pillars, and it enhances the panicle branching, and then it also increases the number of spikelets per plant. And finally, the yield per plant also increased. So currently, we are almost the harvest data is getting ready. We have a large plot trial also we have conducted in the net house to generate seeds for the coming Kharif season field trial. So DST gene, it regulates various mechanisms through stomatal closure, altering stomatal density, and also altering cytokinin metabolism. It provides yield stability as well as the stress tolerance. And we have also started work on the bacterial blight resistance in rice. We have to import this bacterial blight edited rice line from Germany, and the work is in progress. And tomorrow, Reddy sir will be talking about it, so I am skipping this slide. This is another example where in rice, the CKX2 mutation was created by Dr. Satyendra at IARR, which gives significantly higher yield. And this is another example about the nutritional security, that is the work carried out by Siddharth Tiwari, where he has created mutation in banana LCYC gene, which resulted in enhanced provitamin A. And this one, since you know that banana is cultivated vegetatively, so recently he has developed a protocol for using RNP mediated editing. So soon I think he will be coming up with a similar mutation in banana. This is an example in rice where the aroma can be created in non-aromatic rice. This is an example from NIPGR where they have created the glucosinolate-free mustard. So this part I will skip because Madam will be presenting about it. GM, it is considered as non-GM, and fortunately India has also joined the global community in declaring the SDN1 and SDN2 mutants, which are free from exogenous introduced DNA, can be considered as normal plant type. So already these are commercially available. The oleic acid rich soybean, which is available from 2019 in the market, GABA rich tomato from 2020, and the edited fish from 2021. These are already available for the consumer. To summarize, it is a precision tool to create mutation for food security. It is also an important functional genomic tool. We can modify any of the gene elements in the genome itself, and you can easily study its function. So we need not overexpress or introduce foreign gene to study the gene function. So that's why this is very important tool for functional genomic to discover the new gene. And we need more and more people to be working on. As I told one of the slides, 2030 summary of the genome editing work that is going around the world. India is almost negligible in that one. It is not there. So more and more people are required. And Dr. Ajit sir was talking about the patent rights. Yes, we have to obtain the patent rights from the Cortiva or from the original discoverer. And government is making all-out effort. So soon we expect that the freedom to operate will be available for Indian community. So if you want to make globally competitive agriculture, we have to use new tools and techniques, not only genome editing, any techniques that can be used. So finally, I thank the funding agencies and for providing me the fund, and all my students, specifically Santosh Kumar for the work which I presented, and Dr. Abdin for giving me a chance, and Professor SPS Kanuja for listening patiently. Thank you, sir.
Thank you, sir, for this wonderful and exciting talk. Genome editing is a very trendy topic, so listening to your work will accelerate many of us to design our work efficiently and effectively. Now it is my great honor to invite our next distinguished speaker, Dr. Amolya Kumar Panda. Dr. Panda is the Associate Director at Panacea Biotech Limited, New Delhi. He did his PhD from IIT Delhi and worked at National Institute of Immunology from 1990 to 2021. He retired from NII as its director. He has published the very first report on high cell density fermentation studies from India in 1995, and he is an expert on refolding of proteins from inclusion bodies. He was awarded the Young Asian Biotechnologist Prize from the Society of Biotechnology, Japan. He has been a fellow of Royal Society of Chemistry. Currently, he is overseeing the vaccine and biological research and manufacturing activities at Panacea Biotech Limited, Delhi. He has 165 publications, 35 patents, and 7 national and international awards to his credit. I request you to please be on stage, sir. Your thinker words on cellular engineering, the next frontiers of medicine through biotechnology.
Thank you, Professor Abdul. I don't remember how long I have been associated with this department, probably from Dr. Srivastav to Dr. Jain. And thank you for continuing that. I really enjoy again coming. It's like coming home. I really enjoy coming to Jamia, both for biotech as well as pharmacy, and I must thank all of you. So what I'll do is I will put you a dream sequence of what will be the healthcare things in 2050. Considering we have a pandemic now, we are all living in a nice, clean environment. I assume by 2040 there will be no infectious disease. We'll be living healthy. And if you listen to what Professor Tanuja told, eat nice things, prebiotic, probiotic, listen to your grandmother, you will not have any disease. Then what next? Maybe I'll give a dream, and many of you who are young, I'm sure you will realize that. So what I'll do is, to know what will be the future, you must know what has happened in the past. I'll give a brief history just to tell you how we have spoiled our health without listening to our mother's advice. Thank God I listened to Professor Tanuja. The importance of good health, protecting your body. I'm sure during the COVID, you must have had nothing worked. Remdesivir didn't work, the steroid didn't work, even the vaccine after taking 4 vaccines, still it is work. It's not a vaccine. What works is your immunity. And those who have learned and listened to their grandparents, taken care of their health, eating healthy foods in every season, they are protected. That's the future. So I'll tell about the history, because we must know what wrong things we have done in the last 100 years. Then only you can rectify. I'm coming back to you in a minute. Then a new concept is coming. Last 100 years was mostly biochemist. You make drugs. Now the new word is bugs as drugs. Can you use live cells as drugs? Everybody talk about cellular agriculture, green chemistry. It is a green drug. No chemicals, nothing. Live cells, it's a bacteria or a fungus or a human cells. Can they be used as a drug? Then the new concept has come. That's called advanced therapeutic medicinal product. And that is nothing but, I'll tell you that. But when I got this invitation, then I saw I'm talking in between all domains of agriculture. I know Professor Tanuja, I know Ajit. Then I thought I'll tell something about agriculture also to make myself more relevant. I'm sure you'll enjoy. So with this, let's see what is happening. This is how healthcare started for the last 2000 years. 2000 BC, we are eating the roots. 2000 AD, we started using the decoction, prayer, then decoction. Then we started making snake oil. Then we started making pill. Antibody doesn't work. Sulfur drug doesn't work. Now, doesn't work. Go back to the roots. In our Sanskrit, there is a saying, Punar Musika Bhava, go back to your original. That's what we are doing. Of course, this root will be, as everybody is talking, it can be a genetically modified root also. That's important. So this is what happened in last 2000 years. Let's see what happened in last 100 years. We have a wonderful chemistry last 100 years. 1920, it was sulfur drug. Doesn't work anymore. Then from World War II, we have antibiotics. Doesn't work anymore. Then we have biologics. Doesn't work anymore. Now we have biologics with chemists. They are making synthetic drugs. Started doing. We have started now cell therapy. Now the future is ATMP. That is, I'll explain you what is ATMP. It is advanced therapy medicinal product. What is that? A new paradigm of treatment is now coming. If you see, last 100 years, the whole concept, whether it is a sulfur drug or antibiotics or antibody, the concept is kill the bug, right? Whatever it is. But now the concept is changing, and more importantly, after the COVID, kill the body than killing the bug. 100 years back, when the sulfur drug were coming, we thought if you kill the bug, you are clean. Then vaccine taught us It's not about only killing the pathogen, but you have to clear the pathogen. It's like solving a rat problem. If you are rat, just killing the rat doesn't do anything. It smells. That's what anti-vaccine does. And the idea is immunity is the best survivor. So the urban therapeutic drug has come, and if you want to heal the body, maybe in 2000 years back, our body, like our Ayurveda or Unani or Siddh, all were telling take care of the body. Now, with understanding of science, and if you look at what my previous speakers were telling, the body is nothing made up of cell, gene, cell, and tissue. So if you want to heal the body, do a gene therapy, do a cell therapy, do a tissue engineering. Like, for example, you have a liver cancer, you are now taking antibiotics, you are taking antibody. In 20 years down the time, you just grow a liver in the lab and change it. That's it. You have diabetics, you are taking insulin, you just have engineered beta cell implant in your body, it will take care of things. You have thalassemia, just engineer gene therapy, that will work. So these are the things coming up. I'll just give a history of how things are coming. Hopefully you will appreciate that. So the medicinal platform, 4 things are coming. You cure at the gene, that gene therapy. You cure at the cell level, that is cell therapy. You cure at the tissue level, at the tissue therapy. And you combine cell gene tissue therapy, that will be combined. In fact, the 20 products, this is little less. There are already 40 products available US FDA approved, and already 2000 clinical trial going on how to clean and cure gene or cell or tissue. This is what gene therapy. Either you change the gene, I'm going to detail because not much has come, but I'll give one example. You either change the gene or you add a gene or knock down a gene so that the disease doesn't come. The best example is these are the products which has come from gene therapy. You can always check in the internet, but I'll tell you one example which just got approved 2 months back. This is Dept.of. This is for beta thalassemia. The fellow who has beta thalassemia, he has to take blood every week and he will live only 30 years. If you take this injection too, beta thalassemia is cured. This is just developed last 2 months in October. Years. This is real bio. Imagine how much of gene therapy, cell therapy, understanding the gene toxicity has been done. But this is the power of biotechnology. Forget about the cost. People will take care of the cost. The insurance people will tell. But imagine a person who has thalassemia, you take this injection, he is cured for the whole life. Don't you think that is so exciting? So this is the power of gene therapy. Then cell therapy comes. I mean, basically, as I told, cell therapy means take live cell as drug, bugs as drug. But again, is it a live cell or can you engineer the cells? With molecular biology, you have wonderful opportunity. You can play around and you can make different type of cells to do a desired things. In fact, the question is, can cell be a drug? Indeed, yes. You see, think during fertilization, before the whole organ is formed, the cell breathes without lungs, the cell digests without jaws, the cell sends without neurons, cells move without organ. That means the single cell has all capability. If you know how to manipulate, you know how to engineer, you can ask it. And is there an example of these are the advantages? Like it can be a therapeutic, it can be a diagnostic, it can be a combination of everything. Because cell has the capacity to move, to sense, to deliver things. As I give you an example, T cells are nature's best robots. You go to a computer lab, what a robot does. Robot is there, you give some signal, it goes to a place, do its job and come back. Think about the T cell. I'm sure many of you must have studied about T cell during time. What does T cell do? The body is full of naive T cells. They stay. The moment the pathogen came, they sense, they go to that place, kill the damn pathogen, comes back. That is what exactly the cells. And in fact, if you see, cells are already working for us. Cells are working as a drug. Look at the macrophages, they kill bugs. Look at the neurons, look at the tendons or chondrocytes, look at the beta cells. They are already working, so why not use cells as a drug? And most of the cells as a drug, which is used on cancer immunotherapy using culture T cells. Remember, T cell is the protection of the body. And most of the time in a cancer, the T cells are not that much amount in number, so that they take amount of immune response. That's why the cancer cell escape the immunosurveillance. So the best idea is, can you isolate those T cells, tumor-infiltrating lymphocyte, grow them in culture and flush back? If you get 10 to 9, which is the body concentration, you are cured. That's what the immunotherapy is going on. In fact, this is a simple way of treating cancer patient, and this is successful. Anybody who has cancer, if you could isolate tumor-infiltrating lymphocyte from the tumor, the concentration will be 10 to 3, 10 to 4. What you do is, you come, am I audible? Okay. So you can grow them in bioreactor and you can flush them body. This is naive T cells. You isolate the T cells from the tumor, and the population is less. You grow up them, but with genetic engineering and biotechnology, you can modify the T. That's called CAR T cell therapy. I'm sure many of us have done. Basically, you get a chimeric antigen receptor. You allow your T cell to express an antigen in the surface, which is opposite to the cancer, so that the CAR T cells go and direct to the cancer. And in fact, huge things are going on CAR T cells. In fact, these are different versions of CAR T cells coming up now. First generation, second generation, third generation. Basically, this is all about tackling the immune system, so that the immune system is activated to kill the cancer rather than taking it down. In fact, you can check many of them are in the clinical trial. In fact, 6 of them have already been marketed. And most of the time, this T cell therapy was designed to treat cancer, but nowadays T cell therapy has come for many infectious diseases, for maybe for hepatitis and other things. So future is for many diseases. In fact, I have a colleague who is trying to do a cell therapy for XRD or drug-resistant tuberculosis, assuming that he will be able to tackle it. So gene therapy, cell therapy, the next comes the tissue engineering. Tissue engineering is a combination of your cell and a scaffold, which is made from biodegradable polymer particles. Ideally, the whole part of body you can make. You can make skin, you can make wound, you can make cartilage, you can make anything you know if you understand the biology, if you understand the developmental biology, and you provide a proper biological needs so that the cell grow. Please remember, when you grow a bacteria, you grow and divide. When an animal cell grow, it spread, it divide, it proliferate, it differentiate, it vascularize. Then only you get a tissue. Things will come, but probably what people have already succeeded is they have started making all non-vascular tissue. Like you can buy from the market your skin, your wound pipe, your urethra, or maybe vein, you can be bought. Look at these. These are products available in the market. Look at this. These are the first mouse which could make a ear. Suppose by accident your ear is chopped off, you can grow them in the lab and fix them. The same thing is the wound pipe. Suppose your wound pipe is damaged because of cancer, you don't know how to speak. This can be grown in the body, in the lab, and can be fixed. These are all of laboratory products available in the market. Hopefully one day you will make a skin, or maybe a liver, maybe a kidney, or maybe a heart, maybe a brain. Those things will come up. In fact, these are the tissue engineered products which have come to the market. Mostly the keratinocytes and the chondrocytes for knee therapy. In fact, the combined cell therapy product, one which is under very late stage of clinical evaluation, is pancreatic B cells entrapped in polymer particle, so that the B cells are not exposed to the human body and you can take care of the insulin. What will be future? Maybe someday you will like to make a liver, a kidney, something like we change the spare part of our vehicle, like you are changing the silencer or engine or something, you can change your liver or kidney or in lungs if you succeed. And beyond cancer, autoimmune disease, there are many things that come. Finally, because I'll tell you, from the cell therapy common offshoot is cellular agriculture. It's nothing but can you do the same principle to agriculture? Like, can you make milk without cow? Can you make egg without chicken? Can you make meat without animal? In fact, for me, I'm basically a fermentation technologist. For me, agriculture and fermentation are same. Agriculture you do horizontally, fermentation I do vertically. All you do is optimize carbon, nitrogen, pH, temperature, environment to improve the productivity. You improve the productivity and you do that thing. But these are already in the late stage of improvement, like making milk or making egg albumin in each system, or making meat, expressing meat, and maybe 5 years, 10 years down the road, you'll get a burger which is made up of chicken, but it is not from an animal, but from lab-grown meat. And the real challenge is coming of making milk without cow. In fact, many people think, I don't know if time will tell, many people think the last 100 years we have lost immunity because all of us got accustomed to drink cow milk or buffalo milk. We are human beings, we are supposed to milk human milk. And a new concept is coming with, I think, the human milk oligosaccharides. These are the oligosaccharides which are the real immune boosters and improving the innate immunity. By drinking cow milk and buffalo milk, we have lost that immunity development. So what people are doing is either expressing these human milk oligosaccharides like 2-fucosyl-lactose, 3-fucosyl-lactose, many of them, or the other way to make human milk in the bioreactor is you have mammary glands, like I told you, we make organ. You have mammary glands, they are grown in bioreactor. You have one media which the mammary glands will grow. You have induction media where the mammary glands will start making milk. A company has already started milk. The cost and quality will come, but the concept and proof of technology is already established. I think these are the things in future it will come. This is how you make a burger. You don't have to kill a goat or things to get meat. Take the cells, grow them layer and compress them, make a dish-like thing, and your burger is ready. In fact, there are other companies. I think I'm in here. Thank you very much. I really thankful to my teacher and students. I was blessed to have wonderful students who taught me how to do this. That's what I have. And in fact, I have a long stint. I from Odisha, then I studied in IIT, lived most of my life in NII, 32 years, and currently I'm in Paratya Bhatt. You can ask me a question. Thank you.
Thank you, sir, for giving us the glimpse of your wonderful work. We are sincerely grateful for every minute detail you have shared with us. The last distinguished speaker of this session is Dr. Vibha Ahuja ma'am, who is serving as the Chief General Manager of Biotech Consortium India Limited. She is an expert on biosafety and regulatory aspects, particularly with reference to genetically modified organisms, having more than 25 years of experience in the field. She is very well versed in issues related to the Indian biosafety regulatory framework and has been involved in the formulation and dissemination of guidelines. Dr. Ahuja has been actively engaged in the policy issues related to gene editing in India through engagement in the development of policy briefs by National Academy of Agricultural Sciences and member of drafting committee constituted by the Department of Biotechnology. Now I would request Dr. Vibha Ahuja ma'am to please honor us with her presence, and she will be addressing the gathering on the topic regulations for products of modern biotechnology, importance and key components. Welcome.
Thank you, and a very good afternoon to all. At the outset, I would like to thank Professor Abdeen for inviting me here, and thanks to the chair for giving me an opportunity to speak in this session to all of these young students. Friends, you all heard about a lot of innovations in biotechnology happening, whether it was agriculture or healthcare. All my previous speakers spoke about very, very exciting innovations which are in the pipeline, from the growing of the organs to a very innovative crops for the nutritional security and the diseases and so on and so forth. So we all conclude from this, I mean, we are all students of biotechnology, and we all conclude that There is so much happening, and this is all because of the advances in modern biotechnology. So now the main point is that while all these advances are happening, there is another facet to all this, and that is the regulation part, to which I would like to introduce you. It's not a very simple thing, so I'm just going to take you through and familiarize you with what exactly are the regulatory procedures for the modern biotechnology products, and would encourage you subsequent to this presentation to also look at this particular aspect and keep yourself updated, because without the regulation you cannot take forward your products from research to the field or for the benefit of the society. So that's why this topic is very, very important, and very important for students to know. In our eagerness to, in the science, and we are involved in that, we sometimes don't look at this, but this is very, very important, and we all need to pay attention to it. So before I proceed to my topic, I would briefly like to tell you what Biotech Consortium India Limited is. We are a company which was promoted by, in 1990, set up by the All India Financial Institutions and the Department of Biotechnology, Government of India. We are in the interface of research and commercialization, so we take up activities which are required to transfer commercialize the products or the products of research which is done in the laboratory in our national institutions, private sector, and so on and so forth. So if you look at the kind of activities which are listed here, project management, consultancy, technology transfer, certification services, information services, IP management, human resource development, and so on. In the sphere of modern biotechnology, one of the very important areas is the biosafety regulations, about which I will talk about. And we found that this is one area where we need to work, we need to have capacity building, we need to take forward products, and so on. So this became like one of the key areas of operations of BCIL over time. Though when we were set up, our focus was mainly on technology transfer, but we realized that simply giving a technology from the lab to the industry doesn't work. There are lots of other things that the support is required, and that is why our area has expanded, and biosafety is one area. So as in the introduction for myself, the moderator said, we've been working a lot on the area of in this area for almost 25 years, and we have worked with various ministries on various facets of this particular aspect. So very quickly, I'm not going to take much time on this. The modern biotechnology began with the development of recombinant DNA technology in 1975, and that involves, we've already said, but I'm just going to say it with the preamble, that genetic engineering techniques, and of course, my esteemed speakers before that have highlighted much more advances than simply the recombinant DNA technology up to the cellular and so on and so forth. So modern technology, as you have seen in agriculture, healthcare, you heard the application, but more than that, even in the case of environment, in the case of enzymes and so on and so forth, there are lots and lots of applications of modern biotechnology. So in the past, as you saw, though it was very much limited to biotechnology, like, making bread and cheese and so on, and look at where we have headed. We have reached to the level where we can construct the organs, we can construct the plants, and so on. So very powerful technology. But that powerful technology raises in the minds of the people some concerns which have to be taken care through regulation. And also it's our responsibility to bring safe products for the society. So what exactly is the genetically modified organisms, which the products of modern biotechnology are called? It's an organism where we have altered, using the genetic engineering techniques. Now you would say that, after all these presentations, we're going back to the definition. But let me tell you, for the purpose of regulation, the definitions are very, very important, because it is your definition that drives what is going to be the regulatory instruments that will be applicable. So these techniques are through recombinant technology. These are also various terms used, like transgenic. We will also use genetically modified organisms. In some instruments, it will be living modified organism, and so on. So when you carve your path for regulation, you have to actually study the definitions also. So this I am not going to emphasize, because we've all heard the applications. So what is unique, and why do we need to, from the regulator's point of view, what is unique in a GMO? The uniqueness is that now we could introduce genes from any source, bacteria, virus, fungi, animals, and we overcame the taxonomic barrier, which was not there in the conventional processes, and that is how it is different from that. Now, this particular point was taken note of way back in 1975 itself by the scientists who developed it. So the first meeting for the regulation of this technology was held in 1975. A conference like this in SLMR, USA, where they opined that this kind of technology may be misused or it may, because there was very little experience. I'm going to take genes from one organism to another. I should check for the safety before I bring it to the society itself. So first set of recommendations came in SLMR in 1975. But you can understand that the focus was more on the use of technology. It was a new technology, and the recommendations mostly focused on safety of the laboratory workers. Now you can think about like the pandemic when it happened, everybody was worried about the doctors and so on and so forth. But later on, as the technology advanced, laboratory and human health in general, but as the technology advanced, the focus shifted to the end products in addition to the technology with the release of the commercial products. Now, we have biosafety, every country has a national biosafety framework. And why do we have that? Because we want to regulate production and release of these GMOs. In all countries with biotechnology programs, this kind of thing is there to ensure the safe access to new products. And above all, provide a level of public confidence that whatever is coming to the drug in the market is assessed to be safe. When we look at this, this drug has been approved. Everybody was anxiously looking at when the COVID vaccine will be approved and then used. So that's how the regulations are important. So I'm going to take you through there are international agreements, there are various countries have regulations, but we will talk a little about the Indian regulatory system. So in India also, when in 1980s, early 80s, mid 80s actually, the research in biotechnology was initiated. 1987, the DBT was set up. And in 1989 itself, we have been very early movers in this area, and the biosafety regulations came up. And these regulations came up under the Environmental Protection Act, under what we refer to as the rules for the manufacture, use, import, export, and storage of organisms. Subsequently, rules came in 1989. But how do we update ourselves with the advances in research? And that is done through the issuance of various guidelines. And these guidelines is a continuous process. I will show you that. So in our country, every activity related to GMOs is regulated under this particular rules. That activity includes virtually the entire spectrum of activities, right from the research to import, export, storage, manufacture. Everything is virtually regulated. In fact, in the rules, it is written in the scope that new gene technologies will also be regulated. How the implication of that was, I will explain to you a little later. So as the rules are under the Environmental Protection Act, the Ministry of Environment, Forest and Climate Change is the lead ministry for implementation. This is supported by the Department of Biotechnology, which provides all the scientific support for implementation of these rules. And there is a role for state governments in the monitoring. Once their products are approved, there may be some post-release monitoring required, and that is done through the state government. And all this is done by these ministries through what we call as competent authorities or statutory bodies which have been defined in the rules itself. And these include, they have different roles. There is a committee which is advisory, which provides advice on some new technologies like gene editing. When it came, some discussion is required. How does India move forward with this? So RDSC will take the broad areas, whereas the day-to-day application processing is through these three committees. Every institution has an institutional biosafety committee. Institution, those engaged in biotechnology. So your esteemed institution also has an IBSC. So then there is RCGM, which functions in DBT. We have Dr. Shasani, Dr. Chunaswamy, and Dr. Panda, all who have served in these committees, from time to time. Then there is GEAC, which is the apex body. And then we have these state and district level committees. This is just a list. I'm going to leave this presentation here and is available to be used by anyone. So then, you can go to the portals of these RCGM and GAC and look for the text of these guidelines, because I'm just going to briefly take you over what they cover. So as I said, in 1989, the rules came, 1990, the guidelines came, and then subsequently, as you can see, 98, 2008, 2011, then even those guidelines have been regularly updated. So this process moves on along with the research as it moves forward. So this is the 2017 guidelines. Basically, they focus on the containment procedures, biosafety levels, at what kind of containment we should have when you are doing your preliminary work. Then we have the transgenic plants, what kind of greenhouse you should set up. That guidance is provided in this. Field trials, that's a very big thing. So confined field trials, these are called confined field trials because we put in conditions for confinement, and these include guidelines for the conduct of CFTs are in detail, standard operating procedures to be followed, how you should file the application. Then the regulators have also brought out manuals. Food safety assessment. So Indian Council of Medical Research has taken the lead. They prepared these guidelines based on the Codex Alimentarius Commission. All these guidelines are not made de novo by countries. Most countries are members of international bodies. So India is a member of Cartagena, is a party to a Cartagena protocol. India is a party to Codex. Codex is a FAO WHO body which makes the standards for food. So we use there are lots of discussions held there. Some principles are agreed upon, and then the countries use those principles to formulate guidelines so that we are also harmonized with what is happening globally. So subsequently, protocols have also been provided. Then environmental risk assessment is a very major issue. Lots of concerns are flagged in the popular press all the time by various organizations. So a dedicated set of guidance is there. So what we want to, risk analysis framework is there. So what I want to conclude by showing you all these guidelines is that we have a very robust and a systematic regulatory process in place in India. So any product which has passed through this system is absolutely safe to use, and there should not be any concerns in the minds of anyone for using. So for example, today morning you have heard Dr. Painter, his product GM mustard has passed through all these testing using these guidelines, and then only the regulators have approved it. So this is a long procedure. It takes sometimes many years because there have been some delays, but then if systematically followed, actually in 4 to 5 years it can be done. So this is what our plants are there, which already have been explained to you. This is all about genetically modified organisms, and the next range of products and the new technique that we heard such nice presentation is gene editing. So when the progress on gene editing started, the research started, the globally activity started, so India also started thinking, how do we regulate gene editing? So the first thing was to go back to your rules and see the definition. So when we looked at our definition, the definition which was given in 1989, so that definition says, application of gene technique called genetic engineering, including self-cloning and deletion. So which meant that all activities related to gene editing will also be covered in these rules. However, globally the world is moving differently. People have found out that SDN1 and SDN2 categories, as Dr. Vishwanathan explained you, are free from any transgenes, and to take benefit of that, many countries have exempted that technology. So taking note of that, after a 2-year process of consultation by Academy of Sciences and by inputs from all organizations, Government of India also has exempted the genome edited plants that fall under the categories of SDN1 and SDN2 from the provisions of biosafety regulations, which means all those guidelines which I listed, all that complicated process will not be applicable to gene editing. So what is applicable, that we will very briefly discuss. So this notification says SDN1 and SDN2 categories are free from any transgenes, are exempted by MOEFCC, and this exemption has been issued under 1 Rule 20, through which the government is empowered to give exemptions. So the process of this, but however, till the time it is confirmed that it is free from any transgene, that work will in the laboratory will be carried out in the same way as for the GM products. So that means the initial work, recombinant DNA safety guidelines 2017 laboratory work will be applicable, and after that, once you have confirmed that this is free, and how you are going to confirm that also we will very briefly take up. Guidelines for use of gene editing plants, subsequent details have also been brought out by the department. A detailed process has been brought out, and in this process you can see there is a separation. There is a separate process for SGN1 and 2 where there is no transgene, but there is a separate process for SGN3 where there are transgenes and they will be treated like a GMO. So a very detailed workflow has been provided, right from the design of your guide RNA to reagent delivery and what all stages are regulated, and these are regulated by your institutional biosafety committee, and what subsequent to that what is not regulated. So that is why I said in the beginning, whenever we are doing research, we need to be acquainted with what regulatory procedures are so that we follow them diligently and are able to take forward our products in a smooth manner. The guidelines also provide for some information requirements, but those information requirements were very general. So then the department has taken another initiative that we will bring a very clear SOPs. So because India is really committed to use this tool for bringing out new products, new crops to meet the challenges, and that is why all this has been done in a very expeditious manner, and the SOPs have been brought out. Now, SOPs provide for very detailed how when you are initiating the trials, suggested procedure for handling, how to take exemption, when if you have to import, then what to do, and then record keeping. So this is the detailed information. I'm just going to highlight you the bold one. Other than all the research work that you routinely do, most important is evidence to confirm that it is free from exogenous introduced DNA. So all these terms have been very carefully very carefully added. So it's very important for you all to know. Detailed protocol has been shown to show that it is free. The absence of the marker through PCR you have to demonstrate, and the procedure also has been explained. If you want to use any other method, how to use that also has been provided for. So I'm just going to end my not with any conclusions, but on a positive note that you heard all this, and we had a very interesting statement from the Agriculture Minister that we will have the gene edited variety very soon in the farmers' field, and that's a very positive note that the government is supporting all these activities, both from the funding point of view as also the regulatory point of view. And that's a very positive sign for all researchers. And I invite all of you to take up projects in
in a further understanding about the products of modern biotechnology. So I want you to please conclude the session.
I must appreciate the speakers to make me a winner in terms of the time management. You all really stuck to your time and said your things very crisply. So CRISPR technology is working well here. All could edit their text and could convey to the students. I'm sure students that you would have gained a lot of knowledge from the diverse topics that have been covered today. We had beginning with Dr. Ajit Shasani, who talked about trait-based, whether you go for genetic enhancement in classical manner or through genetic engineering, you have to reach the trait, and then that trait could be qualitative or quantitative, and how you link it with ultimately what it is going to deliver. And we had Dr. Vishwanathan, who is from IARI. He talked about the editing part and what has been further elaborated from regulatory point of view by Dr. Vibha Ahuja, that how some part of it would get covered under GM and some will not, and its mechanism was explained by Dr. Vishwanathan. Dr. Panda talked about, he is from IIT, so he talked about all engineering, right from cell to tissue to agriculture. And a dream that he has shown, don't look at it as dream, it's a reality. Reality foreseen, that's it. This is going to happen. Cellular agriculture, my own interest has been and he very rightly pointed out the biggest challenge of milk. But then let me tell you that, you know the fruit jamun, right? So its seed has a protein which is very, very synchronous or parallel to human milk protein. So the answers are going to come out again from plants to take up those challenges, because when you take plants, higher and lower plants, actually it represents the whole thing that occurs in the universe. Animal kingdom is dependent on it. So it is the plant kingdom facilitated by microbes which works, and that's what is coming out from the work that was told. Uh, he talked about AMTP and then its derivatives, which perhaps is going to be the way. Also telling about organs coming out without animals, or the things that had happened in the past, like headless frogs, which tells that rest of the body can be there. If you don't see the face, you don't say who is it, right? So there could be humans, also headless humans, and you have all the organs in them. So it depends on how ethics are going to be regulated, what you allow and what you don't allow. It is in human mind only. The role of police and advocates, Dr. Vibha Ahuja explained very well. So she's making a biotechnology police which can raid you if you don't follow the rules. And she has already told you that what is wrong and what is right, and then it is also evolving. BCIL is doing a great job as a subsidiary of DBT, being a company in itself. So they have to literally behave like a corporate, and therefore the things are coming up so well. So I'm sure that with this session, the plenary 4 lectures which you have just listened, you might have some questions to the speakers. So if there are any questions, please raise your hand, say that question, and also tell that whom are you asking the question among the 4 speakers. Yes, yes. Introduce yourself and then ask the question. You can give a mic, please. Take the mic.
My question is from as we know that local farmers are very prone to fear towards GM crops. So how can we encourage them to grow these crops inside their field?
And non-availability of the social data. So, question is that whether we are involved social scientists to analyze the sphere when we have done the transition technology. We have also that it is very pertinent that when innovating has started, we have to involve social scientists to explain that this technology are not harmful. You cannot reproduce oxygen in the plant and you say that this way. So in that case, the regulatory is the parts. But overall, we have to have the social aspect and see the society mentality and able to disseminate in a positive way how this technology will help and to proceed in. That's it.
But it is also in the popular press, it is highlighted that farmers are affected. This is not the fact. Because if you see globally and even in India, whatever crop was approved, adopted by 95%. So similarly, if you see maize in Brazil, soybean, any of the crops which are approved, whichever country they are in, standard food years, the area is more than production rates are very high. So just a few farmers will be able to do a. That's why I said, popular press, there are lots of things, but you don't have to worry about that. Look at the regulatory system, look at the actual data, because most of these will be, as I said, apprehension based. And also, is lower. In department, there are lots of reports. But if you tell me what is the end result, the end result is within 2 to 3 years, 95% area is under development. And that 5% is because not because people don't want to do because of people, but some varieties in which we are not doing. So this is also very important. We are also in this case. We don't really succeed. So if you don't tell that we succeed, then people will be doing it. So BT is to say, keep a time to say, keep a matter. Then it will be okay. So when you are doing this thing, then you have to somehow you have to be very, very cautious. Very cautious. Tapping the chain or something like that, or bring out the product. So only society will take the basket and press our brand. But how much we do? Positivity.
See, the answer lies in from outreach mode. You have to be now on reach out mode. You have to reach out to the people, make them aware, speak in the language that they understand their fears in their language and answer that fear exists or not, with examples which Dr. Riba was mentioning. So when we say translational science or translational research, it also includes conveying that to the beneficiaries or the end users. And that's where the things are lacking. The reason being political and social, where myths have been created. And always when myths are created, they have to be removed by awareness. So I was talking to Dr. Mahapatra also the other day, and when he was Director General, that let's create a magazine, popular magazine in common language to make people understand what is EM and why it is not dangerous, and where can be the dangers, what they have to be careful about. So that part is important.
What I found is Maharashtra is already approved and we have two of 50 years already, 20 years around but there are these development initiatives in Maharashtra. So there is people efficiency. So they are getting this problem. Immediately are acting on it. So when it is Rajasthan, it is the they are getting because they are not resistant at all. Because it is just initially 5 to 6 years there. So this is a different scenario in two states. And in the case of other things, what only I was talking to professionals, they are saying that a lot of filing is other farmers will be go through side or proceed with other which is not good. We need to be but how can we set that also? And this side both we can do this is a problem. The thing is that what will be the biodiversity? My specific question is how to look after the biosafety issues and also the resistance.
So this found that technology sounds so it is not a problem, but it is a general problem. It's a lot associated with
Any other point or question? Yeah.
Sir, my question is for Dr. So actually, sir mentioned that cows will be replaced by automated meat. So a lot of research is going on in the development of artificial shrimp, artificial fish also. And when it is concerned about meat, so people are more inclined towards artificial meat because of the carbon credits. Some people say that this is a more carbon efficient method, basically. And that is why people and investors are more inclined towards artificial meat. However, a certain group of people say that artificial meat is actually more energy and resource intensive because you require quality nutrients for that. Like, so you need proper cell culture lab, you need proper facility, you need vitamins in order to make sure that the culture has. Yes, sir. So vitamins need to be added, all the nutrients need to be added, the media, cell culture media needs to be prepared, and that is where it's believed that the cost of or to get to artificial meat is more expensive. So I would like to know thoughts on that.
When you look at what you come, it comes very high. I will think. I think it does. But actually not. It's not like what you're doing. One or two. You know, if you want to buy a pencil, you immediately take a bigger pencil. That was so costly. $15,000, I'm very good at that. $15,000 per kilo. You know what the cost of pencil now? $10 per kilo. People that $10, you know, 800 rupees per kilo. Pencil. I have a meter. The cost will not want to develop technology and effect. What is importantly, how do you get? As you told about carbon credit or think of
when you do something in lab, the animal group, think about how much travel time. So travel time is very meaningful. It doesn't create overall. There are bigger polluters in the world to pollute the environment than small things like smoking or burning something. They are minimum. Think about that regarding the cost as the technology develops. Like I feel underground. So we have not only a 3 or 4 by DST. I bought myself 1,200 rupees per dose. Today it is 12 rupees. Population qualified hepatitis B vaccine is 12 rupees. The fellow who is selling 12 rupees today is not doing charity, he is earning money on it. Right? Cost will slowly come as the technology develops. What is important is
इसके बाद कोई क्वेश्चन मत तो अलाउ करिएगा डिस्कशन क्या
everybody was scared of, everybody, many of them don't take vaccine, but during COVID, everybody took vaccine. They were all scared of their vaccine. So when that necessity will come, like we all are talking of people that are there. So think about Indian problem. Every day you see a university who have one time, right? Imagine if everyone was take class and take care of everything to one people, to have that one. So,
Plus, remember, 90% of the meat eaters do not know what is carbon barrier. So it's not, the decision is not based on that. You people can discuss this over tea now. I'm closing this session. Thank you very much.
Thank you, everyone. I request the chairperson of the session to please felicitate the distinguished speakers. First of all, to our first speaker of the session, Dr. Ajit Kumar Shasni sir. I request you to please come up on the stage. Thank you, sir. Trying to felicitate the second speaker of the session, Dr. Vishwanathan Jinnapani. Thank you, sir. Now I request the chairperson to please felicitate Dr. A.K. Panda, sir. And now I request Dr. Vibha Ahuja ma'am to please come up on the stage. Thank you, everyone. Now I request Professor M.C. Darby, Head Department of Biotechnology, to please felicitate our chairperson. Sir, as a chairperson. Thank you, sir. Thank you, everyone. So this was end of session 2 of among the plenary sessions. Now it's time for the tea break. And I would like to request all our guests to please move towards the lobby area. The tea is waiting for you, and while having the tea, you may enjoy the poster sessions made by our budding researchers. They are displayed on the first floor. After the coffee break and the poster session, we will start our technical sessions, which will run parallelly in hall number 1 and hall number 2. Thank you all.
बना रहे थे ना हमारे। बेटा वो बना रहे थे माइक। ये सुनो।
मुस्तफा ध्यान से सुनो। हाँ मुस्तफा ध्यान से सुनो। रीमा मैम मुस्तफा अंदर आओ।
मतलब होगा नहीं। अच्छा इसको चेंज कर।
मैंने कुछ तैयार नहीं किया है। वो मैंने हाँ हाँ उठा लो उठा लो। पेन ड्राइव मुस्तफा के पास है। जी बताइए। हाँ लेकिन आधा कब से आधा
ठीक है, इसका पहले एरिया में आपको
मुस्तफा A very good evening to one and all. Myself, Buksa Shakti from Center for Transient Plan Development, Department of Biotechnology, Jamia Ahmedabad, pursuing PhD under supervision of Professor M.Z. Abdi. Here I'm going to represent an oral presentation. Here I'm going to present an oral presentation on the topic of zinc as a potent regulator to increase viral trichome density and stevia glycosides accumulation in stevia recording. As you all know, that diabetes is a serious health problem nowadays. International Diabetes Federation estimated
पानी का बॉटल दे देना भैया। मैम बिस्किट। सर बुला रहे हैं।
यार वो लड़कियां तो कैन आई जस्ट नो कहां है वो गर्ल्स चाय पीने चली गई जो यहाँ थी बैक स्टेज कौन सा यार सेल्फ रिस्पांसिबिलिटी नाम की कोई चीज होती है या नहीं कहाँ है सब लोग और ज्यादा वो लिस्ट का है जिसकी ओरल प्रेजेंटेशन वाली लिस्ट का फाइल वो ले गए ओरल वाली मुझे तीन चाहिए ओरल वाली
हेलो 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 हेलो
मुझे तीन वो दे we would love if you would stay for that.
प्रोग्राम में स्टार्ट होने वाला है ना स्टार्ट होना तो फिर माइक ऑन ले देते हैं
I request everyone, please be seated. We are about to start the next session. Volunteers are requested to please call all the participants from outside. We are about to start the technical session.
शायद उम्र सब तो ऊपर बोले रहे हैं प्रेजेंटेशन और दिखा मैम को
आप लोग आगे पूरे खाली है ना आगे आ जाओ। Ma'am. Ma'am, ma'am.
Okay, so welcome back. Now we are entering in the first technical session of today, which is based on conservation of rare medicinal and high value plants. To chair this session, I would like to call upon the stage our very own Professor Dept, Head Department of Biotechnology. Now, I would request the invited speaker of this session, Dr. Reema Pant ma'am, to please come up on the stage and share her knowledgeable words on the topic biotechnology from anthropocentric quandary to sustainable solution.
Am
I audible?
Yeah, you are audible.
Okay.
This is forward, backward, and this is pointer.
Okay. Good afternoon, everyone. I have a very difficult task to do. After a long session and with so much of excitement going on, and after a lovely meal and wonderful tea, you've been asked to come back and I've been asked to speak. And this time, winters, I will try my best to not to make you sleep and to see that you're wide awake and you listen to me. So let's try. Good evening, and with the permission of Chair, I would first like to thank Jamia Hamdard and the Department of Biotechnology and Professor Abdeen for giving me this wonderful opportunity to be here today and not only listen to those wonderful lectures, also interact with some of you and have a wonderful time. Topic for today was very difficult to decide because I was asked to speak on herbs. And herbs are one of the most difficult things to speak on because we are still finding a lot of them. We say we know that we have quantified, mapped all biodiversity across the world, across the country, but still every time there is a new finding, every time when we want to find solutions, we go back to nature and we are again looking for something and we find something new. So I've tried to put in something which I have seen through my practical experiences of interacting with certain farmers who've been working in Uttarakhand and also trying to see how solutions can be made. So it is from anthropocentric quandary to sustainable solution. All right, I will take you back into time. And this is important because I want you to observe something. Maybe I'll ask you after some time what it is, so please be careful. So if you look at this is the entire journey of origin of Earth to what we are today. And we see we've passed on billions of years doing this. And if you look at it very shortly, then we find from 460 billion years, 4.6 billion years ago, we have been going on, and man diverged from its primate ancestors only 6 million years ago. And the modern man that we talk about probably came much later. So what basically are we trying to do here? And all these events, by the way, never came without the mass extinctions. Every time something good happened and the nature was full, happy, healthy, hearty, there was a mass extinction. And then again, it started from scratch. So why do you think this must have happened? And it took us so long, 6 million years ago. Then from that, we continue to go on. And if you look at the timeline, you find there's a huge gap. And all of these, our ancestors, are nowhere today. We know that this time the Nobel Prize for Medicine was given to a scientist who actually quantified the entire genome of the Neanderthals. So what it is trying to tell us? It is trying to tell us that nature has refined on genetic composition as and when required according to the surroundings that were according to what is coming. So nature has no
special affections, or nature has no hatred for anyone. Nature is quite impassionate. उसको ना किसी की जरूरत है और ना किसी के बगैर काम चलेगा ऐसा होगा. So it just goes on in its geological processes, and in turn, whoever survives through stays, and whoever is not able to adapt just goes, just vanishes. And this doesn't happen over a period of time. It doesn't happen in 3 months, in 2 months, in 1 year, in 5 years, or putting up a projection and saying, okay, I have projected a model saying that 50 years from now this will happen, so let me make the genetic combination like that, and that is it. So nature doesn't do it that way. And sometimes what we think was the best, if that was the best, would have survived, which did not. Then comes another very interesting aspect in nature, co-evolution. Co-evolution, we know, is something we see every day. Exams होते हैं, टेंशन होती है, पहले तो पढ़ा नहीं, फिर अनफेयर मीन्स की भी कभी कभी जरूरत पड़ती है, तो हर बार बच्चे एक नया तरीका निकालते हैं, नया तरीका इजाद होता है कैसे चीटिंग होगी, और हर बार टीचर्स उनसे वन अप करने की कोशिश करते हैं. सो व्हाट आर वी डूइंग? वी आर इवॉल्विंग अगेंस्ट दिस रूल्स. हर बार साइबर क्राइम के खिलाफ एक नया सॉफ्टवेयर डेवलप होता है सोल्यूशन के लिए, और उसके बाद फिर से एक साइबर क्राइम हो जाता है. So next we again evolve. So everywhere we see this evolution happen, and this is not a staircase. सीधे सीधे नहीं होता. It happens. You have to go back again, look at the roots, then develop yourself and evolve, and it takes a lot of. So if I look at the genetic refinement of how nature does over a period of time, and you look at what basically we say in biotechnology, it's the same. So nature itself has been doing a lot of biotechnological work, basically gene refinement work, to produce what it needs at a given present time in given environmental conditions. If you look at man, and if you see this slide, this is very interesting. All through, we don't see any development happening. But towards the end, after the Holocene interglacial period, suddenly we see that the entire thing happened. Industrialization happened, all civilizations were there, world wars happened, all things happened because we developed a cerebral hemisphere much more and much better than most other animals. And that is probably the reason why we are so successful. Nature gave this to us. It was a bounty that we received and no other animal received. Whatever environmental conditions that would have been there, but we got it and nobody else. And what did we do out of that? Like Maulvi Sahab was saying in the morning that दूसरे को देने का काम तो किया नहीं, but what we've done at the end of it, we've only been taking. And then thanks to all the economists in the West who gave us this idea that everything in his interest for free, so we took everything again. ये सब ले लो, जितना ले सकते हैं. For what? For developing economy. Then the trade-offs came. When economy develops, the trade-offs were we lost our environment. This happened in Delhi also when COVID came. Everybody was shut down in their houses. Suddenly, everybody was happy. Ah, kitna acha environment, clean, everything nice. And the minute we got out, what happened? It didn't take us more than a month to go back to pollution. What kind of intelligent beings are we? Please introspect. I mean, I have no technical lecture as such, but this is more for an introspection to think if we are looking for solutions, then at what cost? If we are looking for economy, then at what cost? I want to stay in Delhi, I want a good house, I want a lakh plus salary, everything at the cost of pollution that my family should suffer, at the cost of having almost a cement jungle around me, at the cost of all these things, at the cost of time that I spend traveling from my office to my home, at the cost that I don't get time except for extra stress. So we have to always trade off somewhere and do this. And one more thing which happened during COVID times was that we suddenly became more interested in herbal. We suddenly became more interested in natural medicine. We suddenly realized that, you know, all these chemical-made drugs are not really so good for us. And we've been seeing a lot of things that are happening besides that. Now, before we come to what happened and where the chemical-based drugs came from and what happened to the natural herbs or herbal-made drugs, let's look at how genes were manipulated by us. Genes we've seen earlier manipulated by nature, took million and billion of years to create what we are today. But in 1800 somewhere, Mendel just out of curiosity, looked at how things are happening in nature and coined the term genes. After that, you look at the excitement. See, we are a very excited soul. We are like small children, always excited to know everything. We always want to know, ab kya hoga, next kya hai? And that is what the serial people cash on in television. Every time in the last climax, they leave you, waiting for the next. But humans don't wait. So the minute Mendel found out what composition the genes are like and how they work and what happens, immediately a lot of people started working on it. And today we have reached a point where, like the previous speaker said, we are actually talking about lab-grown meat. We've already got a pig with a ear made, and we are looking for more solutions. Yes, these solutions are very important for us. We need them because we will need them at the cost of having something alternative to losing our own precious organs. Donate our organs for after death, which is a noble thing. Alternative to have something like a lab-grown thing, something like printing 3D objects as extra parts of your body and just fitting them there. So this is how fast we moved. So from 1865 somewhere to 2003, and in fact to 2020 is hardly any time. If you compare that kind of change which has come in the history of Earth while it was evolving us to what we are today. Now this process of evolution, if you look at corn was only found in Central America, natively. And then after some time, people took it from one part to the other. As human beings moved, they wanted to go, they took their favorite food along with it, and they passed on. Two things happen when favorite foods passed on, or favorite plants passed on, or favorite animals passed on, or accidental animals passed on. We came up with invasive species. Today, India, we are struggling with invasive species. We are not able to get rid of them. Eventually, we've started finding alternatives to say, okay, you can stay here because you are good for our butterflies, you can stay here because you're good because you give us essential oils, but we don't have a choice. I'm giving you all these scenarios because when we come towards why we are wanting a sustainable solution out of biotechnology, the problems are here. These are problems which we can identify all across us. So invasive species became one problem. We have no answer to. Plastics became another problem. We have no answers to. Then we came up with artificial corn. Artificial corn came in, a lot of things. But then again, the problem is that it started impacting our soil, our water, because anything which is not a part of natural system is going to take in extra amount of energy from the system to survive. And the other thing it will do, it will also wipe out the native species. In the morning, one of the speakers spoke about famines which happened. Now, there are two scenarios of famines I would like to tell you. First is the Chinese famine. Once, long time back, during Mao Zedong's time. Mao Zedong was an extremely fanatic dictator. So he couldn't bear the fact that sparrows come and take away a little bit of grain from the cornfields. And he said that all the sparrows should be exterminated. Absolutely, as he said, the Chinese went on hunting all the sparrows from the field, and eventually almost all the sparrows from China were wiped out. Any guesses what happened next? Any guesses? There was an aphid attack, and all the corn, all the food grains were destroyed. Because sparrows were natural predators to the aphids. So when they were eating it, they were also actually taking away all the aphids and securing the food for human beings. See, nature has made a complex web. It's like a structure like this, which is very stable. And all animals and all plants in that entire structure are important. It may not be important for me to have a mosquito. I hate it because it gives me malaria, it gives me dengue. But that mosquito is important in nature for some other work, which may not be mine. So if I'm exterminating something, if I'm changing something's properties just because I don't have an affinity towards it, then I think I'm weakening the web. What if I don't like this structure and I start poking and taking out the middle ribbons of this? This structure will fall because it will get weakened. This is how every time when we are trying to alternate, exterminate, or change any kind of living species, we actually weaken our own web. And we cannot forget that we are a member of that web itself. So if the web weakens, we weaken. If the web falls, we fall. And if the web collapses, we collapse too. So that is why biotechnology is important. At the same time, how, where, and when becomes an issue. There was one person who just asked a question before the session ended, the last session ended, and he had an issue with how the biodiversity will get, affected by genetically modified organisms. Yes, it may get affected. Yes, sometimes there can be problems. We've all had problems. We've had problems with DDT. America never accepted DDT was a problem until it actually became a problem for its own national bird. And then very slyly, it shut off all the DDT in its country and put it all for the third world countries. Again, we just had COP27, it just ended. And we know, again, despite all the push that the developing nations have given, the developed nations will still come up with some alternative plan B to not to share things. So when we take up newer technologies from the West, it is important for us because we have to be at par with them. Otherwise, there are lots of issues that we face, including economic issues. But at the same time, we also need to be very careful when, where, how we are utilizing it in our scenario. While all this development was happening, about 65 million years ago, there was a whole chain of mountains which rose across the globe. In the Eurasian plate, we had Himalayas coming up, we had the Alps coming up on the other side, we had the Urals coming up on the, so almost all of them came up at the similar time. And these are one of the young fold mountains that we know. Now, I want to give you a little scenario of Himalayas. I know most of you must have read it in your geography books. We know about it in geology. But since I come from Uttarakhand and Himalayas, we are all there, whether it's the valley with the lower Himalayas and Ishivaliks, or we go higher up into the greater Himalayas. So it's all there. And it's a vast chain which goes on from Afghanistan and goes up to China, Tibet, Bhutan, and Bangladesh. And in between, it has almost 10 states from India, which are basically Himalayan states. Now, why are we talking about Himalayan states? Because we are rich in bioresources, we are rich in mineral resources, we are rich in water resources. We have a lot of biodiversity. In fact, the Himalayan region, Eastern Himalaya especially, is one of the biodiversity hotspots in IUCN. Then we also have something like a temperature gradient, an altitudinal temperature gradient, which takes us from moist deciduous forests to alpine grasslands to snowline and then just barren rocks and snow. And trust me, almost all these levels have different kinds of trees, different kinds of biodiversity, from which majority of them are aromatic plants or herbal medicines. And some of them are highly endemic also. Now, Himalayas are beautiful. Yes. How many of you visited Himalayan state? Anyone? Which part? There. Okay, you've been to Uttarakhand and Himachal, and in fact, the funniest part is that all Himalayas look different. If you move from Uttarakhand to Himachal, the shades of mountain change. You move to Kashmir, the shades of mountain change, and northeast, the shades of mountain change. Because the geology changes, because the vegetation changes, because the water quality changes, the soil quality changes, the air quality changes. See how little differences can change the entire scenario. So how sophisticated was nature when it developed all these things? It was no human being, it had no cerebral matter, it had no research experience, but it developed very, very minute differences in each one of these different genus and within species also to make it so unique that even no two human beings are unique except for a few which are congenital, we know that. We have a fragile ecosystem. We can't have very heavy industries there. So if we expect good industries to come up there, we cannot have those industries. We have a weak economy. We have a vanishing traditional knowledge because most of the people have moved out of the state. And also because the newer generation which comes in has little or no knowledge about traditional systems which were earlier on there. and you're sensitive to rapid development. Everybody, after, especially post-COVID, if I tell you about Uttarakhand, lots of people from other states have come in, and suddenly they realize, oh, this is a nice place to live because there's no pollution, there's that, so let's buy a land and stay there. And I fear a day which is going to be when we will only find a hill of cemented houses, and the green will be gone. And of course, we have a very volatile geology, which many of us don't know when we buy homes in hills. At least for Uttarakhand, I can say it lies in the seismic zone of 4 and 5. So if it is in the range of 4 and 5, then it can seismic zone 4 and 5 means it can have earthquake, which can be up to 7 or 8 Richter scale, even to 10 Richter scale. And earthquakes don't give warnings when they come. So you can understand how difficult it is. 1991, there was an earthquake in Uttarkashi, and an entire hillock with the entire village fell off the cliff into the gorge, and the village was gone forever. And it happened in the middle of the night, so you can imagine everybody sleeping just went off straight. So it's a very difficult place. One single landslide, extra amount of snowfall or something will just disturb the natural system in which people are living and staying. So it is a challenging world. But during the Stone Ages, there are evidences of this. If you can see this picture, can you see those red pictures here? Can you see these? These are the cave paintings near Almora in Uttarakhand, and these cave paintings are of the Stone Age time. So if people could live then and survive then what has happened now? Why are people migrating out of this place, and why are there ghost villages? We are suffering all these crises. We are suffering an economic crisis. We are still a young state which is under debt. We have a lot of environmental degradation which has happened. There's ecological fragility. We have a lot of anthropogenic pressure as more and more people are moving into the city. And of course, it is going to eventually become an existential crisis. I'll take some time here. These are the issues. These are the challenges. Of course, climate is a challenge globally. We can't help it. Varied weather patterns is a change. But what is it doing? It is impacting the glaciers. It is impacting the rainfall patterns. It is impacting the rivers. If the glaciers are impacted in Uttarakhand or in Kashmir or in Himachal, then the river volume in all the plain areas in India get impacted. And Uttarakhand especially, because we have two major rivers coming out from there, the Ganga and Yamuna, which are actually giving water to more than 10 states across. So that is a serious issue. Then we have pollution problem. You know, last time also there was a heap of garbage left by the mountaineers in Mount Everest. So imagine what kind of species are we? We are the only species in the world who are capable, actually capable of going to the world's most difficult places and creating garbage. I think we really need a round of applause for ourselves because we can only do it. No other, believe me, no other animal does this in the world except for us. This is just straight out of heart. And in fact, 2 days before coming here, I had gone to border district of Uttarakhand called Champawat, where the other 5 districts, border districts of India in Uttarakhand, we had called government school children from villages to that place. So there were about 240 small children from class 6 to class 12. And trust me, some of them had never even seen a city. So for them, that little district town in the border was a city. And they were so excited to be there. Can you all sitting here even imagine not seeing another town? Not seeing a bus, or not seeing seeing a bus but not seeing a train, or not seeing other people. So for them, it was an amazement. So you can understand how difficult and how challenging the pressure is for them. Then we have population pressures, which are more floating than resident. We have less resident population because most of the people go out to work and women are left. If you look at the villages, most of the villages are ghost villages. There is nobody living in those villages. Just a couple of houses which are degrading and nobody. And just fields. Because everybody has moved out from there. And then if you come to cities or better villages, then there are only women and children also study and then move away. So old people and women are the only ones left to fend for themselves in the villages. it's a critical situation. With the forest nearby getting degraded, the animals move into their farm. So if you, it's a very rich belt. Uttarakhand is a very rich citrus fruit belt, and you have all sorts of citrus fruit there. We were talking about fruits and nutraceuticals and vegetation and herbs in the last presentation. They can actually provide it to a lot of people, but there are so many monkeys and there is so much of wild boar menace in their fields that they can grow just nothing, not even food for themselves, because monkeys leave nothing and wild boars take away everything which is underground. So now think of a situation, you are living in a place which is about 2,500 meters high up in the hill. Water is a problem for you, extreme weather conditions are there, you don't have proper roads to live in, and whatever little things that you grow are eaten up by the monkeys or the wild boar. What is this? How do we tackle it? What should we do? So all this happened. Invasive species have taken in. Distribution patterns of flowers has changed. Flowers which were supposed to flower in March or April have started flowering in January and in February. So there is a distinct climate change. Biodiversity is threatened. Traditional knowledge is going, and sometimes even intellectual property just goes, because hill people are simple. They will tell you their traditional medicines without realizing that sometimes biopiracy will simply take place. So there are so many issues that are being there, and they have to be tackled. Now we know that 21 to 2030 is the decade of ecosystem restoration that has been, that is being celebrated by IUCN, UNEP. And this is what I have that I wanted to share with you, that Himalayan ecosystem too needs restoration. As an ecosystem, it needs restoration without compromising development, because people live there as well. So we can't have a disbalanced economic distribution with city people and people living in the plain areas having a higher economic output, and people living in the hills which are just barely making their ends meet. And if this happens, then ecosystem will not get restored. And trust me, if the ecosystem doesn't stay, that none of us stays, because that belt actually controls your climate, your water, and other important aspects. Therefore, it is something which we need to consider. Coming back to the herbs, remember we were talking about herbs. We said that COVID actually made us so aware about having natural products and having artificial things. So if you talk to majority of these, companies have come up, especially companies like Patanjali, which have been promoting. We have already had companies like Dabur and Vednath and Himalaya, which always have been giving us products which are herbal and natural. But ever thought, where are all these herbs coming from? Do you think all these herbs are growing somewhere like wheat and rice, like we saw the output of wheat and rice, which was shown by the earlier presenters, how much we've increased in number? Do we think that all these herbs are also coming in that potential? These herbs either grow around the homes or they grow in the wild. Sometimes they're so endemic that they grow only under certain conditions of temperature and moisture, otherwise they do not grow. So this is one untapped resource which is there in the hills, and that is what is basically the premise of my talk today, that this rapidly vanishing resource is the one which could be a potential economy generator for the villagers in the hills, which are especially women, and also keep the biodiversity safe. And we will come to it where biotechnology is important. So these are certain, this actually is a WhatsApp conversation from a farmer who sent me this. As to, I was asking what are the forest collections you take, because majority of the herbs that we get, especially for the typical herbs, which are very, very significant, we get it from the wild. And see, when you're taking herbs from the wild, imagine when we teach children about the bycatch in ocean, what we say is the trawler goes, और जितना का सामान होता है नीचे, चाहे कछुआ हो, eel हो, कोई मछली हो, घोंगा हो, जो मिले सब उठा लो उससे, and tons of it comes up, and half the animals are dead. So we don't see what animals we are catching when we take a large trawler and just pick up things from the sea. Similarly, when you send some untrained or semi-trained man to the field and say, "Bhai, 5 garu haldi utha ke le aao," he will just go collect everything which he finds or looks and feels it is, he'll pick it up and bring it. Whether it's a young plant, whether it's a flowering plant, whether it is seeding, he has no value to it. All he knows is if I collect certain number and give that certain amount, I am going to get money. So what happens in it? We lose biodiversity. This is where we lose biodiversity. Because then that seed will not come back. Then that genetic composition has gone. So we need to leave some seeds. Earlier on, we had this very amazing concept in India of sacred groves, where we left certain plants. This was a tribal culture, where we left certain plants to themselves and nobody touched them ever. And that is why they became pure genetic, that was a gene pool which was very pure and it just multiplied on itself. But now we don't do that because we know plants can be cut down, plants can be used, it has various uses, so we don't care about it. So like this, all these which you see here are medicinal plants which are collected from the wild, from the forest. So if somebody, and these are many of them are those which are found in snowline areas. So it is about 3,500 meters and above. अब अगर वहां पे जाके किसी ने सारे के सारे तोड़ लिए तो दोबारा कैसे होगा? Imagine snowline is a point where temperatures are very low, so it takes a lot of time to grow. So it will take a lot of time to grow. So if you take away all of them, it will never come back. Therefore, similarly, now these are the ones which have a possibility. Like I told you, there are women farmers who are having problem with animals in their fields. So herbs are one thing which animals don't eat because that doesn't interest them. There is nothing nice about herbs, and especially these herbs that I'm talking about, all of them are very bitter. So the wild boar will not touch them, the monkeys will not eat them. And if they're cultivating it in their fields, they can actually get a lot of money out of it, which will maintain their economic standards also, and they can buy food, they can buy other things out. And it doesn't take too long. These are ones which can grow below snow areas. So somebody who is living below snow areas in warmer regions can grow this. But the point is that are these women sensitized? They are not. So they don't know what they can do. They don't know how to grow. They don't know what is the use of it. They don't know where they will give it. So my this thing here was that can we make a nice supply chain method for them where we do two things. We use biotechnology to make, we give them, instead of asking them to grow it right from the seed, we make callus or we do tissue culture and give them tissue cultured plantlets and allow them to grow, which makes it easier for them to grow. And then also teach them how to not only grow but process it a little bit and send it off to the right place, or maybe give them a contract farming system, something where these could be sold. Otherwise, we sell it. We already, like Jatamasi, I'm showing you. This is again, this grows at an altitude of 3,000 to 5,000 meters. Very nice truck, but again cannot be done without supervision. It is also coming under threaten because it is collected from the wild. Nobody grows it. People are trying, but then again they need to be sensitized. They need to know how to do it, and some technical person has to teach them. So this is where our role comes in as educators, as biotechnologists, to help them out. Similarly, these are a couple of drugs. So these are all drugs which are found at higher snowline area and very, very sensitive, very, very important for. So this is where we need to identify, we need technology, we need sensitization and training, packaging and marketing. So if we can give them this entire package, then women living in these far-flung areas can actually get benefited from it. And now with technology on our side, I think this could be an easier option to work on. Biotechnology, this I need not tell you because this is a map of Uttarakhand where already it has been, this is an old map, basically 8-year-old, but it has these pointers where already we are doing aromatherapy work. So it is not that we are not doing it, but this is actually clustered in the low-lying areas, areas like Dehradun, areas like Haridwar or Udhan, which are flat, plain areas. What my request here is that if some of you would be interested to do this work, but go to the higher hills and actually work on it, which could be a game changer for us because we will get, these drugs are they are very high-paying drugs. So if it is on a contract and if you are selling it properly, one, we get good herbal medicines from whoever makes it, and we can also make enough money and our women become empowered. So that is all for me. If there's anything that I can add. Thank you so much. Thank you so much for being patient, sir, and for hearing me out.
Thank you, ma'am, for this interesting session and for enlightening us on the.
Just 1 minute. I just want to ask, I hope I did not make you sleep and you were not pinching yourself to stay awake. Thank you for that.
Not at all. Thank you for enlightening us on anthropocentric belief. It was lovely having you here. Now the moment has come to add a golden feather in the hat of success. So we are going to invite the selected young researchers for the oral presentation. They will be getting 15 minutes each to showcase their. Okay. So only one speaker is there. Okay. So the speaker will be getting 15 minutes to showcase their research and discoveries. Judges for the oral session will be Professor Shahid Umar sir, Dr. Saima Wajid ma'am, and Dr. Reema Pant ma'am. So consequently, I would like to invite upon the stage the first researcher, Ms. Qudsiya Ashrafi.
Thank you so much, ma'am. Very good evening to one and all. Myself, Qudsiya Ashrafi, from Center for Transgenic Plant Development, Department of Biotechnology, Jamia Hamdard, pursuing PhD under supervision of Professor Anzal Abdin sir. Here I am going to present oral presentation on the topic, auxin as a potent regulator to increase glandular trichome density and steviol glycoside accumulation in Stevia rewarddiana. As we all know that diabetes is a serious health problem nowadays, International Diabetes Federation estimated that there will be 700 million people will be with diabetes by 2045. And India is at the second position after China with the highest number of adults of diabetes in top 10 countries. So diabetic people use different kind of artificial sweeteners and natural sweeteners. So if we see the market of these sweeteners, the high intensity sweeteners cover the 9% of the total global sweetener market coverage. And in this coverage, highest to lowest are the aspartame, sucralose, saccharin, acesulfame K, cyclamate, then stevia and neotem. If we see the market level, after the 2008, when FDA approved the use of steviol glycoside, stevia became a major player in the sweetener industry for using a natural sweetener. And global stevia market was valued at 637 million in 2018 and projected to reach 1,169 million by 2026. So here we can start with the introduction of stevia. Stevia rewarddiana is a native plant of Paraguay from the family Asteraceae. It is known to the scientific world for its non-caloric sweetening properties. Basically, the leaves of stevia contain diterpene glycosides, mainly steviosides 5 to 10% and rewarddioside A 2 to 4%. Stevioside is reported to 143 times sweeter and rewarddioside reported to 42 times sweeter than sugar. Beside its sweet taste, anti-hyperglycemic, antioxidant, anti-cancerous activities are also present in these compounds. And if we talk about the accumulation of these SGs, stevioglycosides are synthesized and get accumulated in glandular trichomes, which are present on the leaf and stem tissues of Stevia rewarddiana. If we achieve an increased concentration of these trichomes, so we can achieve the mass product can be achieved through plant tissue culture technology. Here is a question that why plant tissue culture? Because plant tissue, with this technique, we can explore all of the power of stevia. So plant tissue culture technology can be used profitably for conservation of elite germplasm and selection of better genotypes
containing higher content of economically and pharmaceutically important secondary metabolites. And this technique provides large-scale production in any seasonal and regional conditions without safety and quality-related publications. So, according to this theory, we hypothesized our objective. First was in vitro micropropagation of stevia. Then evaluation of trichome density was done under confocal microscope to evaluate the effect of plant growth regulators on glandular trichome density. Then HPTLC analysis was done to analyze the content of stereocyte and body also. Then GCMS profiling was done for whole metabolome profiling. In the first step, we generate a complete plantlet of Stevia rhodiana from nodal as well as leaf explants under different hormonal concentrations. These are different concentrations like BAP and combination of BAP plus NA which were used for nodal explant, and leaf explants were inoculated because of the property of its embryogenesis because cytokines are known to play a role in shoot multiplication. Then in vitro rooting was done on IA, which is a natural rooting compound. Then optimization was done for whole micropropagation of Stevia rhodiana. Mother plant was procured from CMAP. CIM meaty variety was procured and cultivated at herbal garden of Jamnagar. Then nodal explant after sterilization inoculated on MS, and the shoot regeneration was observed on MS plus BAP. But when we used BAP in combination with NA, we saw an improved shoot proliferation. Simultaneously, leaf explants were also inoculated. Here, indirect shoot regeneration was achieved on the combination of BAP plus 2,4-D. And direct shoot regeneration was observed on BAP plus NA combination. So these shoots were excised and multiplied on the combination of BAP plus kinetin. Then in vitro rooting was done, IA. Here is raised complete plantlet of Stevia rhodiana, and they were hardened and acclimatized. So here we achieved our first objective for the micropropagation of stevia. But when we use these combinations, here we found direct shoot regeneration from leaf plant. But interestingly, when we found indirect shoot regeneration, here we can see that we found some increased hairy appendages on stem tissues. So we analyze these hairy appendages on under microscope. Then we found these structure which was similar like the trichome structure which found in the stevia leaves. Here are non-glandular and glandular trichome also. So if we have introduction about some plant trichome are highly specialized epidermal hairy appendages found on the aerial region of the plant, like on the leaves and on the stem tissues also. They are classified as unicellular or multicellular, glandular or non-glandular, and they play important role in tolerance to biotic and abiotic stress by protecting plants from UV light and insects. And one of the key point of these trichomes are that they accumulate different kind of secondary metabolites like terpenoids, alkaloids, flavonoids, which are economically important compound. These glandular trichomes act as mass factories of the cell. So if we see the base of the formation of glandular trichomes in Arabidopsis, means model plant species, it was found that phytohormones or environmental stimuli can modulate both the onset of secondary metabolites accumulation as well as trichome initiation. So if a cell converts into trichome, glandular trichome mainly, then there can be a huge amount of secondary metabolite can be accumulated here. Mice et al. Reported that in Artemisia, anti-malarial compound artemisinin was increased in the glandular trichome in presence of some different kind of phytohormones. So we also observed stevia leaf samples under confocal microscope to found that our plant growth regulators affect these glandular trichome density or not. This is the control leaf sample. Here you can see we found small trichomes, non-glandular, large trichome which are also non-glandular, and here is glandular trichomes. And the leaf cultured on MS plus BAP, not a huge difference was shown. This is BAP plus kinetic. But yeah, when we use the combination of BAP plus NA, the trichome density was little bit increased. And in the combination of BAP plus 2,4-D, we observed that trichome density was increased in the matter of size as well as numbers were increased. So we found a significant positive correlation between the augmentation of auxin in the medium and glandular trichome density on leaf tissues. So here are the results that in presence of Na and 2,4-D, the glandular trichomes were increased. So thoroughly researched the literature and we found that in Passiflora foetida, researchers reported that in presence of 2,4-D, they also found the increased number of glandular trichome density. And they also reported that after 5 mg per liter of 2,4-D concentration, there was a morphological alteration, just maybe because of herbicidal nature of 2,4-D. So same as we also planned to increase the 2,4-D concentration in our media on which plants were cultured. Here we found some interesting result that in presence of 2,4-D, glandular trichomes were increased. We increased the 2,4-D concentration from 0.5 to 1 mg per liter. Then we observed that the hairy appendages were decreased and glandular trichomes were increased. And further 2 mg per liter, 3 mg per liter, 4 mg per liter, and at 5 mg per liter concentration, we also found some morphological alterations like apinesty of leaves and trichome bunches at stem surface. So we also conclude that in presence of 2,4-D, trichome density were modulated, but at higher concentration of 2,4-D, there were morphological alteration because of the herbicidal nature of 2,4-D. And we discussed that these glandular trichomes accumulate secondary metabolites. So we analyzed our secondary metabolites, especially stereoside and rewardoside, that they were increased or not. We did HPLC analysis to find out the content of stereoside and rewardoside in the leaf as well as stem samples. Here we found a positive correlation between the content and the concentration of different PGRs, in which in presence of Na and 2,4-D, content was also increased. So here we found increased content in the media. For whole metabolome profiling, that not only stereoside and rewardoside A, what effect was on other secondary metabolites like alkaloids, flavonoids, glycosides. So we generate a heat map analysis through GCMS that which showing correlation between the relative concentration of different metabolites in leaf as well as stem tissues. Here is the BLS-BA analysis which identified the relative concentration of the corresponding metabolite under different PGR concentrations. And here we can see in the GCMS chromatogram a clear peak increment in the flavonoids and steroids. So Conclusion is that the center of attraction of the present study was to find out the best concentration and combination for micropropagation of Stevia ruboriana. But interestingly, we found a higher density of glandular trichomes in culture. So the study extended in terms of correlation of PGRs with trichome density and stevial glycosides accumulation. So this developed protocol can be used for micropropagation of stevia through direct as well as indirect from nodal as well as leaf explant with higher yield of not only asges but other important secondary metabolites also. This study reported a significant role of plant growth regulator, especially auxins, which were Na and 2,4-D, on density of glandular trichomes with an improved metabolic profile. The finding of these suitable PGRs could be used through the foliar spray technique under field conditions. So after Bondarev et al. Study in 2010, we are second in the queue. Recently, we published a paper of this crispy protocol on the influence of plant growth regulators with the reported findings of glandular trichome density increment as well as stevial glycoside accumulation in stevia. So, if we talk about the short summary of these findings, is that we found an active correlation between the plant growth regulator, glandular trichome development, and stevial glycoside accumulation. But in future, we are planning for find out the molecular basis behind this active correlation, because in tomato, Zhang et al. Reported recently that ARF3 genes play an important role in the formation of this glandular trichome. So we can also find out a new study. These are the references which we were used. Acknowledgement to an awesome team. Jamia Hamdard for providing a green and peaceful environment and working place. DBT for providing me JRF fellowship. Last but not least, this conference to providing me a platform to share this research work. Thank you. Thank you, Pujya, for sharing such an interesting work with us and sticking to the time. Any questions from audience or from judges? Yes, please. The border side A. Yeah, that's it. The body side A is the key important secondary metabolite of stevia leaves. After stevioside, it is the main component of stevia leaf, which is higher sweeter than stevioside. It is 242 times sweeter than sugar. And yeah, its content is low in stevia, but we are in our lab also trying to enhance this content, because now it is FDA approved, so we can use it as a natural sweetener. Side effects of stevia? No, WHO estimates that stevia did not cause any genotoxic effect. Many research are there, but it is safe to use. Because in the market, many products are there now, and even in our lab, our supervisor and all the lab we do stevia tea party also. So it is safe to use. Press notices are allowed to. It was done in 2008 by Food and Drug Administration. Okay, thank you. Any other questions? Trichomes. As we discussed, the trichomes are the mass factories. They function as a factories of cell, like they accumulate the different kind of secondary products. Thank you. Any other question? Yes, please.
pre-primary stage, we find out that maximum glycosides were accumulated in the state. Yeah.
Does stevia get metabolized in our body?
Metabolized or not in our body?
In the sugar body used by the intestine and blood microbes, and then it goes to liver and excrete.
Okay, thank you. Any other question? Yeah.
My question is stevia safe to use or not? Yeah, it is safe to use. I just
We already discussed this. Please, your question, please.
Thank you.
It was very insightful presentation. My question is, in today's world where very new innovative techniques are coming, can you say that this plant culture technique is an old technique?
Okay, I am agree with you that plant tissue culture is an old technique, but if you will gonna agree with me that old is gold, so yeah, plant tissue culture technique is laborious, I agree. But whenever we talk about conservation of especially plant species, then the first technique which came into our mind is plant tissue culture.
Thank you so much. And now, due to some health issues, Mr. Irfan Bashir Ganai could not join us for the oral presentation. So now I would like the chairperson to please felicitate the invited speaker of the session, Dr. Rima Pant ma'am. Please bring the memento. I request Professor Shahid Umar sir to please present the memento to Dr. Rima Pant ma'am. Now I would request Professor Enzit Ardin sir to please felicitate our chairperson, Professor Shahid Umar sir. Thank you, sir. So this was all for today's technical session. Thank you all for your patience and cooperation. We will be meeting again for our third plenary session tomorrow morning at 9 a.m. Our cultural program will begin soon after 10 minutes break. So thank you all.
Pendrive